Do you want to, Christina, do you want to thank the sponsors right off the bat? Because I cannot see that on my document here. I don't think I'm on the, oh, wait,
It's
I've got it.
page.
No, here we go. Okay.
Okay,
Thank you, everybody, for being
okay,
here. We're back.
hang on,
I'm trying to,
hang on.
are we audible? No.
Welcome, everyone, to the Napa County Farm Bureau Continuing Education.
Recording in progress.
All right. I'm gonna leave one. Excuse me. There we go. Hey, everybody. How's it going? Thank you for joining us today. We've got a great presentation lined up, and we've got some text I'm getting. Zoom link does not accept my credentials. Will Thomas. So maybe behind the scenes, if people are reaching out, please contact Kristen or Christina directly for technical assistance, and they can get you lined up and loaded. Um, I want to begin by thanking our sponsors. We couldn't do this without our sponsors, and so great appreciation to them behind this specific training. Barber Vineyard Management, who's been a full session sponsor, ETS as well, Grow West, and Wilbur Ellis. Without your support, we couldn't do these things, and we've been doing them for 13 years, and you guys have been involved for 13 years, and we greatly appreciate it. A little background on Farm Bureau. I do this every time, but for those of you who don't know Farm Bureau, hopefully everybody on knows Farm Bureau by this point. We're a grassroots organization, nationwide, county-based, and the sole purpose of our group is to protect farming. And everybody on this call, I assume one way or another, you're connected to agriculture. And so supporting Farm Bureau is supporting your future and your present. And so please do what you can to learn more about the organization. If you want to get involved, we would love to have you involved. I've been involved for 16 or 17 years now, which is really dating me. But I've enjoyed it, learned a lot, and there are tons of opportunities that will benefit you personally, in addition to supporting a cause that's near and dear and important to all of us. Beyond that, I want to say thank you very much to everybody at Farm Bureau. Christina specifically, she has done a wonderful job organizing these things this year, and Anahi and Kristen as well. It's been a really solid process, and that would not happen without strong folks behind the scenes doing a lot of work. And Peter Rumble too, our new CEO, fantastic guy. If you have not had a chance to meet him, please make the effort because he's Napa County's in good hands, and you'll feel that way even more once you've had a chance to acquaint with Peter. What else? Today, we're doing this remotely, and so we're going to have some polls at launch. I don't know if Christina, do you want to say a word or two about the polls? They're really important to you if you want your CE credits. It's going to be hard for us to provide them via DPR if you do not hit these milestones during the presentation. So do you want to
And just a little bit about it, there's about 15 poll questions through the 5 hours here today. You need to answer all 15 poll questions. The first question is a verification of your attendance. So we will send that out here as soon as we have everybody in the class, and they will begin during the speakers. So please answer your poll questions quickly, and we'll proceed forward. Thank you.
All right, and now on to the good stuff. This is a tech-focused, training. I try and put one of these together every year, and I'm more excited than I ever have been. I know about the speakers we have and the subjects we're going to cover. First off, we have Matt Clark, who is an assistant professor in the Department of Horticultural Sciences at the University of Minnesota. His focus is dual, both traditional and molecular plant breeding approaches to develop improved fruit cultivars for cold climate farming, and focused within that, studying the genetics behind unique resistances common to pests and diseases, while improving the eating experience through the development of exceptional texture and flavor. Those are really, really important things for us across the world of wine grapes and other crops as well. The goal, one of his goals, is to develop new apple and other fruit varieties with multiple resistances so that growers can reduce the amounts of pesticides applied during the season. He's a project director for Vitis Gen 3, which is a USDA NIFA project sponsored through the SCRI and focused toward disease resistance. Matt, as I stumble over my words, please take it away. Very nice to meet you virtually.
Well, good morning, everybody. Make sure that we're on the right screen and you're saying the right thing. Someone can confirm.
Yep,
Slide
we
text
got
up.
it.
Okay,
Perfect.
great. Well, thanks for the introduction and the welcome here. I was reflecting last night. My first engagement with Farm Bureau was when I was a kid. I attended the Iowa State Conference, and I think that year Elizabeth Dole was a speaker, if that gives you any indication of my age. I was a kid, and I remember going to the trade show and seeing cellulose-based packing peanuts for the first time. So clearly, some things that really stuck out in my mind that I still remember today. Well, my goal today is to introduce you to our Vitis Gen 3 project. You're fortunate that I won't be presenting much of any data in this talk, so any poll or quiz questions that come up won't be related to that, so you can thank me later. But it is a bit of a deep dive into our grant project because I think it's important that taxpayers like yourself know where funding goes and what our goals are in this research project. So my aim is to introduce you a bit to our team, our main objectives, but also provide you a bit of historical and scientific context for why we're doing the work we're doing and how it's having impacts across the whole country. It might surprise people in Napa, but the rest of the states all do have wineries and are producing grapes and supporting their own industries. Although those are small, they're really important to our local economies and supporting rural agriculture as well as agrotourism. So without further ado, I'll just hop right in. Maybe I got to get on the right screen. Okay, so our project is part of the Specialty Crop Research Initiative through NIFA. This is funding that comes through the USDA. And the way that these projects get funded are through a lot of support from industry who have a critical role in reviewing the proposals when they're submitted, but also in the development process. And I'll point out a few people that you might actually know when I pull up a photo of them in a moment. And our project has national, regional, and state importance, and we aim to hit key target areas of this grant program. That includes plant breeding, genetics, and genomics, which will become very obvious as I talk about the project. This is our key focus. And we address pests and diseases, and powdery mildew has been the disease that we've been focused on the most over the about 15 years of this project. Although regionally, there are other pests that come into play, and that'll become more obvious as I go through the talk this morning as well. We also look to improve productivity and profitability, and mainly that's been focusing our work with horticulture and ag economists to help develop better tools for marketing, especially as we talk about developing new varieties that aren't Chardonnay or Merlot or kind of the key Vitis vinifera products. Our new products need to find a place into the market, and we need to figure out how to best promote and advertise those so that we complete that feedback loop of planting new varieties and then selling those products. And then new innovations in technology, and I'll show you some different examples of those that we're implementing in our program, which we think are pretty cool for doing the research that we do, but also may find their way out into producers' fields as well. So our project is funded for 4 years and with over $10.3 million. So it's quite a large project, one of the larger ones from this program. It's actually hard for us to spend that much money in the 4-year period just based on our alignment with the different fiscal calendars used by the USDA and universities. So we'll probably roll into a 5th year. We have 13 funded institutions and 29 different PIs, or in this case, faculty researchers or folks from the USDA plus collaborators. And then we have a really important stakeholder advisory panel. And maybe you can recognize a few faces in this photo from California. For example, Franca Gobler from the Table Grape Commission is here in this picture, Donnell Brown from NGRA. We have other growers and producers from around the country, and then representatives from Gallo, from the USDA, and so forth. So we have a strong team of advisors as well as our scientific community who supports this project. So as I briefly mentioned, our project has been going on for roughly 15 years. We're in our third and perhaps final round of funding with the VitusGen name. And the title of that project is Completing the Grapevine Powdery Mildew Resistance Pipeline from Genes on the Shelf to Sticks in the Ground. And we came up with this terminology to hopefully demonstrate that by the end of this project or throughout this part of the project, we actually take the information that we gained in the first decade or so of the project with understanding genetic control for powdery mildew resistance to having varieties that are out in testing. And we are being successful with that. So I often start presentations about grapevines asking this question: how many grape species are there? And since we're virtual, I don't really have a great mechanism to get responses from the audience, so I'll just tell you. But if you look at the picture on the right, you can probably get a sense that there's at least 4. But in fact, there's quite a few more than that. There's about 60 species of grapevine found around the globe. We break them into 2 groups, U. vitis and Muscadinia. And U. vitis, or what we normally just call vitis, sometimes people say vitis, whatever you want to say is fine. These have a base chromosome number of 19 pairs or 38 chromosomes, and that's important for breeding purposes. And about 30 of those species are found in North America and 30 species in Asia. However, the one that you're most familiar with is Vitis vinifera, which is a predominant cultivated species, is from Europe and Eurasia, which is kind of the oddball in the group. It's the one that had been selected by humans for flavor and aroma for winemaking and table grapes. But we have all of these other species that offer other unique traits. The muscadine grape, if you're from the southeast part of the United States, you'll certainly know about this wild grape that's been brought into cultivation. It's different in that it has 40 chromosomes, so 20 pairs. And this is a really unique one because it has multiple disease resistances with it, including the RUN1 powdery mildew resistance gene. And this is an important one for you to remember about your CEUs as we're going through the presentation. RUN1 comes from muscadine, and grape breeders decades ago started making crosses or trying to make crosses between these different species, Vitis vinifera and muscadine grape. And because they have different chromosome numbers, that presented some major challenges. But fortunately, through persistence, they've been able to incorporate this RUN1 as well as some downy mildew resistance into Vitis vinifera research and breeding programs that we're using today. And I'll talk more about that as we go along. So where are these grape species in North America? Primarily, they're in the east and distributed across the north. Where I live, we just have one, but it has a broad range. It's called Vitis riparia. And you'll notice that California, of course, has its own native grapes, and these species move into Mexico, one species in the Caribbean. But there's a wealth of genetic information and resources here because these species evolved here in North America. They've evolved with our native pests: powdery mildew, downy mildew, black rot, phylloxera. That list goes on. And these species are well adapted to the different soil and climate and weather conditions, as well as day length. And we think about developing new varieties of grapevines that have resistance genes. Grape breeders need to turn to these crop wild relatives to bring in those genes. And where I'm from, in Minnesota, hopefully you see that purple star. If you're not familiar with where Minnesota is, we're right in the smack dab middle of the north. Vitis riparia has played a huge role in our breeding program for over 120 years or so for cold hardiness. And through breeding efforts, we've been able to develop 11 cold hardy wine grape varieties that support our region. You might be surprised to learn that Minnesota has about 80 licensed wineries. I know that pales in comparison to where you are, but that supports over $100 million of economic impact into our state. About 30% of that is in tourism, 30% in wine, and the rest in grape production. So that's really important for our farmers and for getting people out into rural Minnesota. Wineries have really become community centers, especially in small towns where people celebrate life and get to drink wine. And here's just a picture on the right-hand side of Vitis riparia growing up a blue spruce tree at my parents' house. Wild grapes are weeds. I think we probably all know that. In Minnesota, I say if you walk outside and walk 100 to 200 yards in any direction, you're going to find a wild grapevine. And that's cool. As a plant breeder, that means that we have access to these wild plant materials that we can use in breeding to bring in new traits. Some quick, fast facts about Vitis is that wild grapes are dioecious, meaning that there's male plants and female plants. And so in the wild, you need both to produce fruit. And because of this, they're also wind pollinated, which helps to get that pollen from the upper parts of trees where the male vines are onto female vines, probably somewhere in the canopy. We all know that grapes are important to wildlife. Most of the grape species interbreed easily, except for the exception I talked about already, and that they're really important to humans. Vitis vinifera in particular, because of its fresh eating quality, turning those into juice, to wine, to raisins, and wine has had a long storied cultural and religious significance that continues today. And not a surprise to this audience, but economically important as well, with a market size of over $10 billion globally. The cool thing about having all these wild grapes in our backyards, literally, is that plant collectors, plant breeders, genomicists, geneticists, plant nerds have been out collecting these wild plant materials and putting them into what we call germplasm centers or repositories. And there's two of those in the United States. One's with UC Davis, I think it's in Winters, and one at Geneva, New York. And this is our kind of a holding place and clearinghouse for all the genetic diversity of grapevine. Of course, it's not everything, but several thousand selections are entered at both of these locations, which are used for breeding purposes. You can think of them as the gene bank for grapevine. And plant breeders like myself turn to those to use them. These types of collections are also held in places like China, which I've had an opportunity to visit, as well as in France, where we're really interested in using this plant material to advance viticulture and wine production. The other thing that we have is just a nice resource is that we have places that clean up vines. You're all familiar with grapevine viruses, and we have national network of clean plant centers that are used by breeding programs, by nurserymen to be able to make sure that the plant material is virus free. And these kind of go part and parcel with the repositories in some cases. I think grape breeding is pretty cool in that it emerged at the same time that other controlled crosses and where plant breeding was really taking off in the middle of the 19th century. And part of this is because people were curious and learning about learning about germ theory, learning about movement of pollen. Animal and plant breeding was really taking off with just animal husbandry, not really the science of it yet, but folks were interested in developing new things because they could see that happening in nature or within their own crops. Something kind of interesting happened with grapevine, though, is that non-Vitis vinifera species were being spread around the world by plant collectors. And when they did that, they brought pests into Europe. So phylloxera came in, downy mildew and powdery mildew came in from the United States. And so plant breeders then really had to scurry together and come up with some solutions to these diseases. And they knew that the American species were resistant, if not tolerant, to these pests and diseases. So they started crossing them with Vitis vinifera. And at the end of the day, long story short, we ended up having rootstocks, sorry for the misspelling, rootstocks for controlling for phylloxera, and that changed the production system for how grapes are produced worldwide, but also hybrids that went into production. Those fell out of favor in Europe, but in North America, hybrid grapes, especially in the eastern part of the US, have been kind of the mainstay of how we've been able to have grape and wine industries. So we've been using these wild crop relatives in our breeding process. It's no different than what's being done in tomato breeding, in potato breeding. Name a crop, all plant researchers are turning to wild relatives because of their local adaptation to climate conditions and soils, but also for resistance to pests and diseases. So I've listed a bunch of them out here of what we're using grapevine wild relatives for. And I'll just hit a couple of highlights. So for pests, we've been studying insect resistance in my lab. For phylloxera resistance, what actually infects the leaves in our region. In our region, cold hardiness is important. So we've been looking at climate conditions and using our locally adapted varieties. For quality, we know Vitis vinifera is actually the contributor to a lot of that. But if you've tried cotton candy grape, that unique flavor comes from Vitis labrusca, which is another one of our native species. Disease resistance, you're going to hear more about, and I've already kind of hit on that. And then soil adaptation is really critical. So we know we're trying to grow grapes all across this country, all around the world, and the soils are highly variable. And the native species can contribute to things like pH tolerance, architecture of those fruits, and so on. I know this slide is hard to read. It's supposed to be animated, and I couldn't figure out how to do it on my new computer this morning, so you're stuck with what you got. But in the background is a pedigree of one of our varieties called Itasca. It's shown here as MN1285, the number we give it before it gets a name. And the key takeaway from this is on the background, it kind of looks like a bowl of spaghetti of all of the different contributors to Itasca. But if we boil it down, there are 6 different species that are contributing to this variety. And from those species, we get things like powdery mildew resistance, cold hardiness, phylloxera resistance, downy mildew resistance, lower acidity than the native grapes, which are often high in acidity, and high sugar, which is also a trait of many of our wild species. So not just Vitis vinifera contributing anymore, our breeding programs really lean into a number of these different species, many of them from North America. So that's a bit of an overview of grape breeding in general, a bit of history, very much a crash course. And the next thing that I want to cover is about our grant in particular. So you know what we're doing, where we're going, and hopefully learn a little bit about the technologies that we're using as we go forward. So I list the objectives here, but I'm going to talk about them one at a time. So nothing that you need to remember here because we'll touch on each of them. So our first objective is genes on the shelf. And our aim here is really to take the knowledge that we've gained from the first decade or so of VitusGen, where we've been mapping the location of resistances to the grape genome, but diving even deeper into understanding the genetic code and what candidate genes lived there. In early days, we didn't have the resolution to get to the individual genes. And now with cheaper sequencing technologies and improved approaches just over time and computational power, the price of genome sequencing going down, we can now actually choose candidate genes to test. And our labs are being quite successful in doing that. So what we're able to do is identify individual genes in the region and then start testing them. And some of the ways that we're doing that is to take a Vitis vinifera plant that's susceptible to powdery mildew and insert into it a powdery mildew resistance gene and see if that is the one that actually causes resistance. So we would infect it with powdery mildew and see if it's still susceptible or if it becomes resistant. And that's our way of gauging if the candidate is actually functional. The opposite way of doing this technique is to take a powdery mildew resistant plant, one of the native ones that I've been talking about, and then knocking out or removing the candidate gene and then infecting it with powdery mildew. And if it goes from being resistant to susceptible, we know we've got the right targets. And you might wonder, why are we actually doing this step? It's one, because we really want to know that we got the right gene. And by having that information allows us to have better DNA tests. It helps us to better understand where to go in our wild germplasm and our germplasm collections to look for more genes like this to use in breeding. But our aim is not to develop CRISPR-based varieties. Our goal is to use CRISPR technologies to actually answer the research questions. We know that right now the grape producers, growers, wine drinkers aren't interested in CRISPR products. And so we're just using that technology because it's available to better understand the science. So our achievements through year 2 were to actually start investigating a number of these candidate genes. And the left-hand column is a list of them. They have names like Run and Ren. The top 2, Run 1 and Run 2, come from the muscadine grape. Remember that? That's an important one on your quiz. And Dario Cantu's lab at UC Davis has been focusing on that. He's identified candidate genes and has worked on actually knocking these into susceptible varieties and is doing the testing. And you'll see other colleagues and kind of our targets for where we're working on some of these other powdery mildew resistance genes. So in addition to RUN1, we also have REN1. And just briefly, REN1 comes from Vitis vinifera and Vitis sylvestris, which are the Eurasian species from which Vitis vinifera was selected and the one that we all love. And the cool thing about resistance genes, we often think that there's just one version of it. That's the one we're going to work with. Well, REN1 is kind of unique in that when they screen through a bunch of different wild accessions of this, I see that REN1 gene is there, but there's several different forms of it. And that's really helpful for us as plant breeders because these might have different associations or different capacity to control different isolates of powdery mildew. And our goal is to determine which of these is really the best ones that we should be moving forward in our breeding program. And having diversity is important. It shows to us that there certainly was likely powdery mildew in this area and that we have evolution for that pest. So I don't want to go much deeper into this, but the work on this specific project is being conducted at UC Davis by Manon. Two other ones, REN6 and REN7. I don't know if I said what the REN stands for. R just stands for resistance, and the E stands for Insenula and Ericepha nicator, the scientific name, and then they each just get a number. And as we find new genes, they get a higher number as we go along. So this is a colleague, Adon Dekny, who's at University of Maryland, Eastern Shores, probably not on anybody's radar for doing grape research, but Adon has been studying grapes and perfecting this technique for doing candidate gene expression in grapevine. And his technique actually takes some individual cells, convinces those grapevine cells to grow into embryos, just like within a seed. And then he can do gene editing techniques with those, grow out those plants, and then infect them with powdery mildew. So kind of a specialized technique. It's an approach that his lab and one in Florida have been doing for maybe 15 to 20 years now. It's slightly different than some of the other gene editing techniques, but he's been very successful and he's been a fun collaborator on this project to have. So now that Adon has produced these plants, they've gone over to UC Davis. They got shipped there, I believe, late spring, and Melanie and the team and Dario Cantu's lab is inoculating those with powdery mildew and looking at gene expression. We get the right genes, candidate genes that underlie the REN6 and REN7 region. There's likely dozens of genes in that region, and usually only one of those is the one that we're targeting. So it's a little bit of trial and error, and I'm hoping to report more back on that as we go forward. So just quickly moving into objective 2, this one has kind of the cutesy name of tools in the toolbox. This team is based primarily out of Cornell University and the USDA at Geneva, New York, and they're working on developing DNA markers and tests for us to use as grape breeders. They are developing a lab technique called Blackbird, which is shown to be very successful in what we call phenotyping or scoring plants for disease. And I'll show you that in a second. And then vineyard imaging, which is shown on the right. This is a prototype of what we call phytopathlobot, so plant disease robot. And you'll notice that this one's held together a little bit with lab tape, duct tape, tin foil. It looks like there's some pink flagging tape. Maybe not the most robust for us to be using. So as a prototype, we've decided to actually move forward and mount all of this equipment into utility vehicles that can drive through the vineyard and capture images. It was designed for powdery mildew, but you'll see in a second that it's just a high-powered camera. And so anything that we can score from a digital image, especially using computer-trained models, will get us data that we can use in plant breeding, but also maybe someday also be used in your vineyards for scouting for diseases, estimating crop yields, and the like. I think it's important to stop here for just a second and talk about what is DNA-informed grape breeding and how we use DNA markers. There's a couple of different ways that we do that. One is using DNA markers to confirm identity of plants. probably all have experience growing grapevines, and when they fruit for the first time, you say, hey, that's not Merlot, that's Pinot Noir, or that's a white grape and not a red grape. Those mix-ups happen, and DNA tests can help us understand that. It also helps us do paternity testing. We have these big pedigrees like I showed you, and we can actually go and find out if the plant breeders got the information right. And we don't use that as often or for validating cultivars. But more importantly, we use DNA testing. You can think of it like 23andMe or individual target genes for selecting which parents we use in crossing. And this helps to improve our efficiencies in breeding to know that the gene of interest, like powdery mildew resistance run 1, is in the parent that we have. And then our philosophy often is that we kill the plants or cull them that don't have the traits of interest. I mean, on the opposite side, we're keeping the ones that have all of the traits of interest, but plant breeders, our job really is to kill plants and find the few survivors. So it's a little pessimistic to put it this way, but our goal is to kill as many plants as possible to find the best ones. So we use DNA markers. I think instead of reading through this slide for the sake of time, I just kind of reinstate that we have DNA tests, 2 different types in our program that were developed by our team. One is to screen for individual genes or the region where all those candidate genes live. And some of those are for powdery mildew, downy mildew, phomopsis, fruit quality traits like flavors and colors, phylloxera, flower sex, and seedlessness. So the types of individual DNA tests we can do serve all the different types of grape breeding programs that are occurring in the US. And if you are doing some of those activities, these tests are or will be becoming publicly available through a service provider that we use in California. So these are individual genes that we're testing, but we also have these genome-wide markers. We gave them the name called RAMP-seq, which I don't expect you to remember. But basically, this is more like 23andMe, where we can have the whole genome covered and get information that can tell us about those individual genes that were just on the previous slide, but also give us better tools for doing genetic mapping, which is its whole other discussion, which I won't talk about today. I think it's amazing that over the lifespan of our VitusGen project, so about 4.5 years as we developed this specific technology for grape, we've done almost 84,000 samples, and that includes all of the plant material at the repositories. So now we have genomic sequences for all of these wild species that can be used for genetic resources. So people often think or ask, like, what's the impact of using marker test? If you look at the upper lighter colored purple, I think you might be able to see my cursor. This is just an example, but in any given year, our breeding program makes thousands of seeds. But for simplification, we'll say we start with 1,500 in the first year. And as we go through this process, we start killing plants. And so the size of that circle represents the population and gets smaller and smaller. And hopefully by years 5 to 10, maybe there's 2 to 10 grapevines that came out of that original 1,500 that we move forward to become a potential new variety and make clones of and start testing, making wines and that sort of thing. But when we moved to making or using DNA markers, in my program we started in 2015, we started testing the parents, testing the offspring, and that allowed us to change some things. We were able to make way more seedlings every year. So we went from really making about 3,000 to somewhere between 7,000 and 10,000 seeds. And then we use markers, which allows us to move out into that first year planting about 1,000 seeds. So about the same as what we were doing before. But those 1,000 that we move forward, we already know have powdery mildew resistance, or they are seedless for the populations that are making seedlessness. So as we go through this timeline, the idea was that by the end of that 5 years, instead of having maybe 2 to 10 top-notch grapes that might advance into the breeding program for more testing, maybe it's 10 to 20 or even more, because we've invested on the front end of only moving forward the plants that have disease resistance. And that's all thanks to this team for developing the marker tools. The other kind of cool thing that they've developed and have really implemented at a full scale is this system called Blackbird. It's a robot that takes a leaf disc and arrays them onto these trays. Those trays are then inoculated with isolates of powdery mildew. And then the robot takes images over what I believe is a 10-day period to look at how the disease progresses, which gives us information for understanding disease resistance. And this image tries to capture what the system is doing. If we start over here with the square, the computer model has learned the difference between leaf hairs, veins, and then fungal hyphae. And so it's able to quantify where the fungi are within all of these different pixels on this leaf disc. So at 2 days, there's 10% infection. At 4 days, 29%, 6 days, 85%. And in this case, almost 100% infection by day 9. So this leaf is clearly highly susceptible to powdery mildew. When you do hundreds of these, we can see the variation and see the disease resistance genes in action. It's an awesome technique, and now it's become highly automated and is a commercial product that actually is for sale. They've taken RGB imaging and now are using techniques for hyperspectral imaging. Basically, instead of just looking at red, green, and blue, we can look at other parts of the spectra, things that we can't see, including infrared. And the reason to do this is because these fungi, although we were seeing them as blue in the previous slide, many fungi have their own spectral signature. So there might be certain wavelengths that allow them to fluoresce. So maybe they show up orange under a certain light, or are detected more easily with something like this. Unfortunately, the second bullet point here is that they produce 2 terabytes of data per tray. So tons of data are required to get this information over that 10-day period. And if we can identify, say, that powdery mildew has a specific spectra, we could reduce that data load and only use one wavelength of light as opposed to the whole visible spectrum and the extended spectra. So really looking forward to seeing what we learned here. Also, this technology can be moved out into the field with the field-based system that I'm going to show you. So the phytopathoblot or PBB robot just isn't cutting the mustard. So we've moved to sticking it to equipment that our teams already have in the field. So we've deployed them at USDA and Parlier with the breeding program there, at South Dakota, at Minnesota. They're already in New York, and I believe one is installed also in West Virginia. And what does this thing do? It drives up and down the vineyard and captures images. Here's an example from the vineyard at Minnesota, and you can see that we actually captured phylloxera in the upper picture, and we see some damage from 2,4-D in the lower picture. So from these pictures, other things that you can probably see just real quickly is that we could see a bunch of berries. So we can measure those berries, we could count those berries. It's just going to take some machine learning models to do that. So really excited about this technology coming out of Dr. Yu Jiang's lab at Cornell. So ostensibly, this has been a breeding program for 15 years, and I haven't really talked about plant breeders yet. We've only been learning about pathologists and some other techniques and gene editing, but we do have, oh, a poll just popped up. I don't know if I should wait for a second so you can do that. I'll give you 20 seconds. All right, that'll probably stay up until you close your own. But our breeding program has also been, our breeding programs across the country have been working on developing disease-resistant varieties for decades. And some of these are now in plantings to better understand how to manage these plants. I think a key takeaway for you all today is that a disease-resistant plant does not mean that it is no spray. We've all probably learned through COVID that pathogens evolve quickly, and that's no different for plant pathogens. And so fungicides will need to be used on disease-resistant vines. This was definitely demonstrated in apple for apple scab, where some VF resistant apple trees were planted, they were only successful at maintaining that resistance for about a decade because people were not out spraying and managing the pest. So disease resistant does not equal no spray. And our goal in this project is to come up with recommendations for extension specialists to be able to have the knowledge to share with growers, to train the growers themselves. And we've established two plantings, but in one location in Geneva, New York, where we're looking at some of these different varieties that have been developed. Arundel out of New York. There's a New York line. Sorry, trying to find my mouse. With a number, La Crescent from Minnesota, which is downy mildew resistant, and then Chardonnay, which is a susceptible kind of spreader here for powdery and downy mildew. And then we have some different lines, these coming out of New York, that have stacked resistances. So different resistance genes have been bred into them, which might behave differently. So our goal is to look at which fungicides are successful with managing the pathogen, preventing evolution of new races that could overcome the resistance, but also looking at biopesticides that could be used that maybe have lower impact on the environment. So we have a bunch of different breeders and geneticists who are working on this all across the country, including Luis Diaz at UC Davis, Sumaira Riaz down at Parlier, and then a handful of others that you may or may not recognize. But this really does show you that grape breeding and grape and wine production is a national effort. And all of these states where we have these breeders have historic and emerging wine production. So these breeders are working on advancing disease resistance. We're testing these vines in multiple states when possible. We're using DNA testing. But I think most important for us to remember is that each of these locations has their own critical needs. So at Minnesota, for example, cold hardiness is first. If the plant can't survive the winter, it doesn't matter if it's disease resistant or not. And so a student of mine, Doug, shown on the right-hand side, and the hat is doing a study to understand the genetics of cold hardiness, where he's freezing these buds and seeing at what temperatures they freeze. And then the team on the left, Laura, is a new graduate student in our team at Minnesota. Now I'm going to blank on this guy's name. Is a PhD student at Cornell, and this is the PI from Cornell showing us the imaging system as they're out collecting disease data in the field. And then each program has to focus on things like quality. So color development, fruit quality are really important, especially with climate change and red color development. We're interested in downy mildew resistance and black rot resistance in the east, because when we release powdery mildew resistant varieties, that means we're reducing the number of sprays that are used. And when we do that, these other pathogens aren't going to be targeted by those sprays indirectly or directly. So we want to make sure that we understand the genetics of resistance for these so that we can start combining powdery mildew, downy mildew, black rot, and other resistances all into kind of what we jokingly call, and sometimes seriously call, a super grape that can handle all of those pest pressures with fewer fungicides being used. And then we have a project that's looking at the genetics of rooting. We often joke that grapevines, if you look at them, they'll grow roots when you make cuttings, but that's not true for all of the species. And so there's a project down at Missouri that's trying to get a better handle on why some of the native grapes and hybrids just don't produce roots when we make cuttings from them, which is also true for muscadine grapes, one of their major challenges for commercialization. And then finally, this is our last objective, and it looks like I'm right on schedule, so we'll have time for some questions, is, do you like to fish? If my dad was here, he would say, I don't have time to answer that, I'm out fishing. Grapes on the table, and this is where our economics team comes in and helps us to better think about marketing our new products that are disease resistant. We realize that we're gonna have to compete with these resistant and tolerant products against the top grape varieties that are out there that you guys are all familiar with, especially in the East, where we're trying to crack into some of these new markets. So our goals are to understand consumer preferences for these new cultivars and within the context of information overloading. And to simplify, that means when you walk into a wine shop and you're presented with thousands of labels and products, how can we get consumers to find and identify our new products, what captures their imagination or interest so that they try something that maybe is more sustainable. So we're looking at eye tracking, which has been used by my colleague Xingyan Yui from University of Minnesota to do that. And she came up with this idea of also connecting that with EEG or brainwaves to see what is lighting up on labels and packaging that consumers are relating to, with the intent of improving label design in general, but more specifically for what additional information can we put on a label when we know that labels have lots of info, that there's federal mandates and state mandates that go onto those, what additional information could maybe hit that, or maybe it's a sticker, still to be determined. So here's just a quick example of what this looks like, the eye tracking and the, I can't even think, the helmet, I suppose, that goes over the participant's head as they're going through the study. And we developed our own mock labels with our own brand that we're using. These are just some experimental stimuli that people would see. And it's kind of probably hard for you to see on your screen, but each of the green sort of stickers on the labels say different things. And we're still working out what those are using input from our industry advisors. So some say sustainably grown or organic, disease resistant. And we're trialing this on wine grapes, table grapes, and also on raisins. Oops. And then basically almost to the end, but I just wanted to highlight and thank some sponsors of our project. This past summer, we were able to send a large number of our graduate students and some of our early faculty members who were new to grapevines come out to California and actually see grape production at a large scale. And one of them described it as a sea of grapes, which I think is pretty apt. And in the upper left-hand corner is our economics team looking at some real packaging. It looks like it's a cotton candy label. They're in some of the vineyards at Sunworld, visited the USDA at Parlier. And so a big shout out to our industry advisors and teams who helped sponsor that trip, really introducing about 23, I think, project participants to what great production looks like when it's not, an acre here or there, but a sea of grapevines. And it was really informative for our teams to get to do that across sectors, including raisins, wine, and table grapes. Extension and outreach are a huge component of any of these specialty crop research programs. And when we started this project, our goal was to reach 3,000 people. And we've far exceeded that, and we've been quite successful. So hopefully you've heard about us before today. And if you haven't, I'll give you the links on the next page of how you can stay in touch with us. But over the past 2 years, we've had over 30 academic presentations, 44 workshops, field days, grower meetings, and so forth. 24 out of the 29 PIs participate, so we're all doing this in every state. And then we've had over 20,000 digital interactions, which was quite the surprise to us. This is where we thought, oh, we might get 1,500 of these, but we have social media, we have our own website, we produce a blog, a newsletter, and then we rely on some other sources to help get information out at all the other participating universities like Appalachian Cornell, Wine Business Monthly, that help give us a highlight of our story. And so finally, if you want to stay in touch with us, we're at vitusgen3.umn.edu, or if you look up vitusgen3 on social media, you'll see us on Facebook, Instagram, and X. And our own grape breeding program at Minnesota is found at enology.umn.edu, and my email is listed there as well. I would say that I am actually no longer the grape breeder at the University of Minnesota. I've handed those roles over to a colleague of mine named Soonli Tay, and I took over the apple breeding program at Minnesota. But I'm still, of course, involved with Vitusgen Project as its project director for at least maybe 2 to 3 more years. So without further ado, thank you for your attention. I appreciate having an opportunity to speak to you. And if there are questions, now would be a great time to ask those. Thank you.
Thank you so much, Matt. There's some discussion in the chat box, and if anybody has questions that they want to address audibly, please raise your hand and then we'll pass the mic. No questions? Well, that's a sign of an effectively delivered presentation. That was
Or
really
not.
interesting stuff,
That
and
could be the case.
it was a great overview. I think that was fantastic, and I may have follow up for you via email that I won't address here, but yeah, if anybody has questions, speak now or forever hold your question. We're gonna give you a minute or two, and there you go. Matt's got his email in the chat box. You can grab that and maybe hit him up directly.
Yeah, certainly welcome to do that. And this will allow people to get another cup of coffee and really wake up for the rest of the day.
Yeah, thank you. No, that timing was perfect, and that was a really insightful um, uh, presentation message you delivered there, and we greatly appreciate it. Let's stay in touch. Hopefully, we're always anxious to establish connectivity between research taking place other places and Napa and beyond. So I'll follow up with you, and hopefully we can do that.
Sounds great. Thanks again for the opportunity. And I know you guys are going to learn a lot today.
All right, take care, Matt. Thank you.
Yeah. Just a little housekeeping note. If you are online and you have two devices open right now, viewing the seminar from two different places, please make sure that you are only using one device to view the seminar. Thank you so much. We have to make sure that we monitor that for the Department of Pesticide Regulations requirements. Thank you.
All right. Do we want to take a break so people can do whatever they need to do? Or hi, Alexa, are you ready to rock and roll? We can just cruise into the next presentation here. How about that?
It's up to you guys. I'm ready, and I'm also okay to wait a little bit if people need a break.
Well, I'm excited to hear what it is you have to say, and I know everybody else is too. So, without further ado, I'll announce you, and then you can kind of just just rock and roll from there. I need to
Perfect.
find your bio, which I have right in front of me here. So I'm gonna work through that, and then we'll go. Uh,
Perfect.
so thank you for being here, Alexa. A little background on Alexa. She is a viticultural research scholar at North Carolina State University. In her current role, she works with North Carolina grape growers and North Carolina cooperative extension agents to provide extension education materials for uh, the best viticultural practices in the regions and conducts grower-driven field-applied research. She received her PhD in horticulture from Washington State University, where her research focused on alternative pest and disease management tools for grapevines. And we've covered this subject, although differently historically, and I'm myself really excited about the technology that Alexa is going to address as a tool within our IPM attack on fungus, because we need alternatives to extend the life of the things that we're using currently, and potentially eliminate some of them that are not as um, agreeable with biology beyond the vineyard, in my opinion. So thank you, and I will pass it over to Alexa.
Pretty cool. Well, thank you for that introduction, Eric, and thank you for inviting me to speak with you. I'm so excited to be here virtually. I believe everyone can see my screen. So we'll just kind of roll with that. So again, my name's Alexa McDaniel. Thank you, Christina. I'm with NC State now, but previously, I finished within a year now at Washington State University. Was this UVC light research? That was my big dissertation. We also covered some other alternative management, but the UVC light was the coolest. Also, it's so fun. Many graduate students don't get lucky to get such a neat project. I did. And this was under the vision of Dr. Michelle Moyer, who I'm sure maybe you've heard research about. Also, this work has been recently published. That QR code, if you use your phone, it should take you to the link. It was published in the American Journal of Enology and Viticulture. And also, this work was funded by the Washington Wine Council, or wine. So just a big collaborative effort. It was also an effort with Dr. David Goodori with Cornell University. And we also worked with Oregon State University Professor Walt Mahaffey. So it was a kind of a good collaboration between universities. We used it in Eastern Washington to manage powdery mildew. And then we also did investigate some fruit quality. So very brief overview. I'm sure you guys are all very knowledgeable of grapevine powdery mildew. But the main takeaway that I wanted to remind people of for the biology of grapevine powdery mildew is that it is an obligate biotroph, meaning that it colonizes the surface. 95% or most of the fungal body is on the outside of the when it infects a green tissue. So that makes it really susceptible to environmental conditions, like how canopy management opening up creates airflow, allows sunlight in to reduce your grapevine powdery mildew or to manage it better. And so that was kind of the key takeaway for using light, UVC light, which is very condensed, very powerful tool. So that is why it has been successful for grapevine powdery mildew, is because of that biology. So to give you perspective of Eastern Washington vineyards climates, we do have, it's a very hot and dry summers. And so even with that, we do have grapevine powdery mildew is one of the main pests for vineyards. I believe our growing degree days are a little bit less than Napa. And also we do have significant, well, not significant, but we do have lower precipitation. We average about 6 inches of rain per year. And so another note from Napa County vineyards is we do have very dramatic diurnal shifts, and we have very lengthy summer days. So we have about a 17-hour day at the peak of summer. And then our diurnal shifts can be pretty dramatic, like ranging from 35 degree Fahrenheit to 47 degree Fahrenheit. So that fluctuation, even though we do have fluctuations that would, reduce powdery mildew, it's still a problem just because of microclimates within the canopy. And also growers still need to spray for that just as a protective. And so with that environmental in mind, how grape growers in Eastern Washington apply their fungicides is targeting the critical window for cluster infection. So typically the fungicide application is, early shoot growth all the way to 3 weeks post fruit set, sometimes a little longer depending on the environmental conditions seen at 3 weeks post fruit set. But it will be, early sulfur applications, and then at that critical window time, which is that 2 weeks pre-bloom to 3 weeks post fruit set, you'll use your most efficacious products. And so those are the systemics, the ones that can translocate throughout the plant. And then typically at the end of the season, growers can get away with not applying a fungicide. Okay, there we go. So, so what is ultraviolet C light or UVC light? Well, it is a short, highly energetic wavelength. It is a part of that that electromagnetic spectrum. It falls in like right outside the visible light range. There's 3 UV wavelengths. You have UVA, UVB, and UVC. UVC is between that 280 to 100 nanometer, and I believe that's a poll question. But in between that wavelength, the wavelength that I am familiar with and that is currently used for a lot of disinfectant and for agricultural studies and for powdery mildew and for other, different research that is happening with UVC light in a pre-harvest pest management scenario is that 254 nanometer wavelength. You will read and see other wavelengths of UVC available. I am not very familiar with that wavelength. I know there's 222 that's supposed to potentially maybe be a little safer for humans. I am not aware of that. This is a wavelength that I was used, and it is a little harmful, so always PPE, but it is not an old tool, or it's not a new tool. It has been used in hospitals, in post-harvest storage facilities, decontamination. It's used in lab hoods to disinfect your hoods. So we know it's a powerful tool. What is new about UVC is how we are using it. This is a great picture that was given to me by Dr. David Goodori. It is Al Michaeloscopy, who was one of the pioneers with David Goodori in creating this pre-harvest UVC apparatus to control powdery mildew. And now they used that machine to control powdery mildew, but they used it during the day. And to see a reduction of powdery mildew, they resulted in these odd russeted, very leathery looking grapes in that right-hand picture. And so the dose that was needed to control that powdery mildew was very harmful to the plants. So, and this was conducted in the early 90s. So then it was put on a back burner, on a hold. They knew it could kill powdery mildew, but it was also harming the plant. And then in 2016, there was a suthopathrin doctor, and he figured out that if you applied grape UVC light at nighttime, you could deliver a dose that was not harmful to the plant, but it killed the pathogen. So the key to using UVC light in a pre-harvest application, such as for grapevine powdery mildew in vineyards, is that dark period. And this is because powdery mildews, they have this robust photolyzed mechanism where they can repair themselves from UV damage with blue light or UVA light. So during the day when you're applying that UVC light, basically you have to apply enough UVC to bypass that repair mechanism. But if at nighttime, when that repair mechanism is off, you are just completely bypassing it. So that allows you for that lower dose. So since that discovery in 2016, Dr. David Goodori has proven, that it is efficacious in controlling powdery mildew for strawberries, I believe for some hop plants. There also have been looking into other pathogens, but the grapevine powdery mildew one has been the one where they have seen a lot of success with it. And so our question wasn't to, answer does powdery mildew or does UVC kill powdery mildew, because we do know that, is how do we integrate it into a field setting? How do we integrate it into Eastern Washington vineyards? And what are those best management practices? So to do that, we built a prototype. These are the two prototypes that I designed and built. The first one on the left, we lovingly called her Captain Marvel, but she's very rickety. She was pulled on a trailer system. And then it was in all designs for these, they're designed as an over-the-row unit to go over, the grapevine and basically surround the vine in light. And inside is, very highly reflective material that bounces off the UVC light, containing the UVC, allowing more time for the plant and a higher dosage. So again, the prototype one was this pull behind trailer. It was pretty hard to, maneuver, but not too bad. The material I built with it was not great, but we did come up with the second prototype, same design. It was a little bit shorter in length. It was on a 3-point hitch that was pulled by our tractor. This one was great as it was elevated, so you could kind of move up and down within the vineyard, and it just allowed a little bit more flexibility. It was easier to drive. Our lights were powered by a generator. The light bulbs themselves do not take that much energy. That QR code in the middle, it does take you to a white paper that we did write that lists the parts, the materials, what we used to build this unit. There is like robotic units that are available and other things, but this UVC light, you can affordably build this, and we kind of are hoping growers will try to build this and maybe, improvise and build something better than what I had, because I was just a graduate student building this and I had someone weld it for me. It wasn't perfect, but I think with some ingenuity of growers, we could really, it could turn into something. Also those lights, they're like just, wired very, just like at any electrical ballast. So if you are handy, you can build this. Also, we did calibrate this to deliver at 200 joules per meter squared, and I'll go into that. And we did use 24 light bulbs just to give you a reference. So how we calibrated it is we used on the top right is what we used as a calibration tool. I will admit it was hard to find a good UVC calibration tool, like as a cheaper. The one that I have listed here is an expensive one. When we tried to use some cheaper models, they never worked, or they were kind of they gave a little bit different readings. They were also in different measurements. So this one I know is reliable, but it is expensive. And so maybe, with UVC light taking off in medical fields and other things, there'll be some better calibration tools. But how we calibrated it to make sure that we're delivering the dose of 200 joules per meter squared. And that is on the high end of the dose. Typically you need 75 to 200 joules per meter squared to manage grapevine powdery mildew. We wanted to do the higher end dose, and you can go a little higher and not harm the plant. But that one was like a dose that was very effective, and we knew that it wasn't going to harm the plant. So you basically do like measurements throughout your housing unit. You do it at the fruiting zone where your fruit is, because that's where you want to target, and you get a reading, facing left at 0 degrees, 90 degrees, and at 180. And then you go at each foot for the length of it, and then you get an average. And that is expressed, that irradiance measure is watts per meter squared. And then the dose of UVC is expressed as joules per meter squared, which is the watts per meter squared times your time. And then you can figure out how fast you can go and what you should be. We calibrated it to go about the typical speed. We were trying to get 3 miles per hour, but I think I was only able to go 1 mile per hour. If you add more bulbs to the light, if you extend the length of it, you can go faster. But the relationship between irradiance or brightness and the dose, so that cumulative quality of UV, dictates the limits of the array design, particularly the UVC dose that can be achieved at practical speeds of travel. So it's just kind of playing around, building, adding more lights, and that can determine how fast you can go. There is a really cool prototype that Cornell has built within the last year or two. You can see that they doubled their lights. So mine was only 24 bulbs. Theirs is 48 bulbs, so they did 2 layers. Mine was only just the one, and I believe they can go faster. And this one, they built it with material was fairly light, where they didn't need the tractor. They're able to pull it with just a mower, which is really neat. We just had a tractor on hand, so that's why we used the tractor. And of course, PPE is extremely important, just like any other of your fungicides. You want to use UV rated face shield and eyeglasses because the most damaging part of UVC light is definitely to the eyes. If you are not careful, it can blind you. So you just want to be protective. And you also want to just cover all your skins and gloves, or just clothing. It doesn't seem to penetrate clothes. You don't want to be like inside the housing unit. Um, and just be protective. Protect yourself just like you would any other fungicide. I would feel like it's the same risk, if not less, with UVC light. If you measure UVC light outside of the unit itself, there is very low UVC, measurable UVC, like standing by it. It's really all concentrated within it in the unit. So being around it is fairly safe. And so that was the machine and how we built it and what it was. But we're now the experiment. Like, why did we build this machine? So we wanted to know how we can integrate UVC light treatments into the Eastern Washington grapevine powdery mildew management. And we wanted to do different nighttime application intervals of UVC light at the higher dose of 200 joules per meter squared and apply it at different timings throughout the season and compare that to standard fungicide program for Eastern Washington for grapevine powdery mildew control. And so we did 2 timing trials. The first timing trial was using UVC light at different intervals just in the early season, so before the critical window period. And then the second timing trial was replacing all of the fungicides that would typically be applied with and use only UVC. So season-long UVC, that was our only management practice for controlling powdery mildew. And so with the early season, we in Eastern Washington, we do have like it's a fairly low risk period to swap out different alternatives just because of how that pre-bloom weather patterns in Eastern Washington are characterized. Though we do have significant, like there is precipitation, there is a little bit humidity that would happen within the canopy that would allow for a grapevine powdery mildew outbreak to occur. We do have like the colder, low time, nighttime temperatures that do keep infection at bay. And also you have less canopy, so there's not so much of a microclimate holding things in. There is some good winds that dry off the plants very quickly. So we figured this was a good time that we could, try an alternative product such as UVC, and that would reduce the amount of fungicide sprays you have, but then you could focus your synthetics right at that critical window period. So we applied UVC from this 7 to 15 centimeter shoot growth, so early shoot growth, to 2 weeks pre-bloom. Our treatment for that was that we did in 2020, we only applied weekly UVC, and then for the following years, we applied it twice a week. So twice weekly. And this was informed by the results that we saw. We decided to increase the frequency, adding that twice weekly. We compared those 2 UVC treatments to our standard fungicide spray and an early season unsprayed. So that was unsprayed up until bloom, and then a season long unsprayed. And we decided to not do any in-season canopy management practices just to try to increase and really create a suitable environment for disease. So we then collected, so how did we assess how well the UVC worked? Is that we visually rated biweekly leaves and clusters from bloom until harvest as a percentage of visual powdery mildew. We then converted that to an area under disease progress curve, AUDPC, which I'll be saying, and the statistics given throughout the presentation are going to be referencing that. So we did convert that because it gives you a good cumulative disease throughout the season. So it really combines all your ratings and shows you how that disease progressed. It gives it nicely condenses and summarizes how disease progressed through the season. So talking more about weather, in 2020 and 2021, we did have extremely warm years and very low precipitation. And so that led to a low disease pressure year. And I have that box on the right-hand side for the precipitation because that critical window period, the 2020 and 2021 are in gray and black. And you can see that there's just like flat lines there. That means that there was absolutely no precipitation through those times. So we had very low disease. And then when you look at 2022, which was a high disease pressure year, it had very stark precipitation increases. And so we actually had disease that year. Looking at 2020, when we look at foliar disease, and these graphs again, AUDPC, the treatments are listed below as well with the raw data, which was the average disease severity. And then the graphs, and I did list the percentages there just to show you what the disease was looking at the end of the season. And kind of like give you an idea of how much disease was out in the vineyard. And the unsprayed is that red. And then we have the same colors that correspond to the early season, unsprayed yellow, weekly UVC purple, blue twice weekly, so tighter interval. And then the fungicide program was in gray. And you can see here that all of our early season UVC treatments did reduce foliar disease compared to that season-long unsprayed control. In 2021, again, a low disease pressure year. We did not have too much statistical difference, but you can see with those percentage, that is the end of the year season foliar disease, and it's pretty low. Like on our unsprayed control, we had 16% in our foliar. So it's just a low disease, but you did see the trend that the UVC did, early season UVC did reduce disease. Now in 2022, when we did have high disease pressure, you can see on this left-hand side, we have a fairly high y-axis, and here you can see it's 250 and then 3,000, just to kind of give how much, and these percentages down there are much higher. And this again is on the leaves, and we did have statistical. So if everything was reduced, so the season-long unsprayed. So applying the UVC did help. And then when you look at clusters, again, 2020, low disease pressure year, there was a difference, but we had 4% cluster disease, so very low. But there was a difference if you with the UVC. 2021, we had absolutely no disease. If you did not spray in 2021 in Eastern Washington, you did not have grapevine powdery mildew, which is very inert. So your sprays weren't needed that year, but it's hard to predict when you are planning your spray schedule for the year. And then in 2022, when we did have that high disease pressure year, you started to see those treatments separate, where the early season unsprayed was higher than our fungicide, but the same as our UVC early. And then it kind of trended towards a pattern here where as your intervals tightened, so weekly versus twice weekly, so a tighter interval, had reduced disease, not statistically, but they're starting to see a trend. And we did have really high cluster disease in 2022. We should have extended our spray program, even in our fungicide program, which was at 25%. That was just due to we should have continued, but we wanted to end at 3 weeks post bloom when weather patterns and what we were seeing, we should have continued. But it was cool to see that UVC early season could be a potential way to reduce your fungicide sprays while still providing adequate control. Also, this early season trial did show us that sometimes you can do nothing early season and still get adequate control as long as you're targeting your synthetics and your really powerhouse fungicides at that critical window period. And so moving on to the season-long UVC treatments. So again, the same type of treatments, except twice weekly, which was the tighter interval, and then the UVC, weekly UVC was compared against a fungicide spray, and then a full season unsprayed. And that was applied from early shoot growth all the way to 3 weeks post fruit set. And we did not do any canopy management because we wanted to induce disease. You can see in 2020 and 2021 with those low disease pressure year, we were hoping for some Ds to be able to prove that UVC was doing something. Again, same data collection, took disease severity percentages and then converted those to the AUDPC. And additionally, what we did for the season-long treatments is that we wanted to look at berry quality. So what happens if you apply UVC, which is a very high wavelength, like much more extreme than sunlight, but it is at a very short interval. What happens to your yield, to your Brix, your TA, to your pH, your phenolics, because we know sunlight does help increase those phenolics. And so we did do a Adam Harbertson method, which is a berry skin dissection. So we removed, and this was in Chardonnay grape, so we didn't necessarily, it's white grape, we wouldn't be pressing on the skins, but we wanted to see if there was some differences. So you make a extraction out of your grape skins. It's an iron reactive, reaction. And so it is your total iron reactive phenolics and then your total iron tannins. But just to give you a little insight into if there is any differences. And these graphs set up very similar to the first trial, AUDPC, and then the disease throughout the year. I do have in that table your end of season percentages just so you can see, what was happening. But in 2020, and this only does have the weekly because we didn't apply twice weekly in 2020. There was no differences. Our fungicide program did work. And so that was a little disheartening to see. There was a trend that UVC did reduce UVC, and you could visually see it in the canopy. We just couldn't statistically see that. And so that is why we decided to add the twice weekly UVC in the following years. For 2020, we do have no difference in the foliar. So no in the leaf, disease. It was just a very low disease. And when we moved to 2022, which the high disease pressure year, we really start to see some neat trends where UVC did reduce compared to the unsprayed control, but it wasn't as much as the fungicide program. And then also as you are tightening your intervals, so twice weekly trended towards better control than the weekly UVC. And so it did reduce compared to the unsprayed control. And I did put in the application period here just to show that if we did go past 3 weeks post-bloom, we might have had better control, but we did stop. And that is when disease started taking off, is after that application period. So in perspective, and looking at cluster disease, 2020, very low cluster disease, no differences there. 2021, there was absolutely no disease. And so, yeah, we had really pretty fruit that year. And then in 2022, there was, again, that trend in the clusters. So we have our unsprayed was the highest, and then we have weekly UVC, and then twice weekly UVC, and then our fungicide program that did perform the best. These numbers, again, are really gross for end of season. I'll show some pictures just to visually show the differences. But this is when we ended our final fungicide spray, and then it started to take off, and then final UVC, again, taking off. So we should have continued that, hindsight. And probably a grower would not have stopped when we did. So looking at those berry quality, we did, this was for 2020 and 2021. We have the total yield, the cluster weight, the berry weight, number, titratable acidity, total Brix, pH. There was no change within those. So when you compared UVC to our standard fungicide program, there was no difference. But in 2022, we did see a difference, and that was not due to the UVC treatments, but it was due to the severity of the powdery mildew on the clusters. You can see this is the way how like disease increased. So fungicide was lowest, then twice weekly, then weekly, and then season long. And then that also is how like the cluster weights and the number of berries per vine, it also decreases. So that tells us that is very typical of grapevine powdery mildew disease influence over those metrics. And then when you look at Brix or pH, as you go down when disease increases that way, then you also see an increase in pH in Brix. Again, a very telltale sign of effects from powdery mildew. And this is showing visually and also why we should have continued on with our treatments, because you can see when infection started and really like developed and affected the clusters. So full spray, not the prettiest, I will admit, but you still have like full clusters. And then as you go to twice weekly, it's still a pretty pulled bin. There is disease everywhere, but the berries are bigger. And then as you go to weekly, you can tell that infection set for our earlier and so much reduced, and then our unsprayed was just like there was nothing on the vine. So UVC was doing something. We just needed to continue or maybe integrate it in a different type of treatment. And then when we look at tannins and phenolics, we did see that it was inconsistently affected through the years. In 2020, you will see that the weekly UVC, and this was milligrams for fresh berry weight. So we took the berry skin, phenolics, got a reading for those, and that was the milligrams that were out of the fresh berry weight. So again, in purple is the weekly UVC. There was higher, and they were statistically higher than the fungicide standard. So we did see that increase. Then we go to 2021, which was again a hot year. We did not see any differences between the fungicide standard and the weekly UVC and the twice weekly UVC. So it was inconsistent. And then when we look at 2022, which did have again, high heat or a high disease pressure year. And we did cherry pick the like uninfected fruit for this measurement. So any undiseased cluster or berry from the cluster we used for these measurements to try to get rid of the influence of powdery mildew. And when we did that, we did not see any differences with the tannins and phenolics. So that tells us that there's probably not enough time that where the berries are actually like where it would be affected. Because when it passes over a grapevine, it's about 4 seconds of UVC light that grapevine receives. So not enough to influence those. Also, I think it tells us that environmental factors are much more of a factor for those over UVC light. There should be maybe a little bit more experiments. We do have some work going where we are using HPLC and trying to identify the phenolics to see if there is not a quantity difference of phenolics, but a quality difference. And that should be coming. We did get results back and we're going through that. But so far it looks very similar. So I feel safe that it is not affecting the berry quality. So we believe that UVC can be a complementary tool for grapevine powdery mildew disease management in Eastern Washington and potentially in some others, but I think it should be a regional experiments for each person, each area, because environmental is such a huge factor in powdery mildew. And while not always statistically significant, that consistent trend in reduction in powdery mildew disease as a result of weekly or twice weekly UVC indicates that the incorporation of UVC into a vineyard IPM program could be an effective alternative for powdery mildew management without compromising that fruit quality. We do believe that with some canopy management we can increase the UVC efficacy by exposing the cluster during the critical window time. And so just how opening up that canopy improves the spray coverage, opening up the canopy will improve the light coverage. And so maybe it really wasn't that we should have continued with UVC. At that time, we had very dense canopy. And so maybe that was an issue why we saw that dramatic increase in diseases, because we had big canopies. This picture over here just shows like you can't see the fruit in there. So maybe that light was not getting to the fruit. So there is future studies, which I'll talk about, that are happening in Michelle Moyer's program at WSU, where they are exploring canopy management options with UVC light. And also, I think it's like our early UVC applications just in the early season, I think does help, provide another option for integration for it. And I did mention that Eastern Washington does have a very long daytime, so 17 hours is quite a bit, and you do need about 4 hours after your application for UVC for it to be effective, and that might not be feasible in some of these very large scale, vineyards that are seen in Napa or in Eastern Washington. So early season would provide that time where the daytime is shorter, so the nighttime is longer. So it provides that needed dark period that helps make UVC effective for powdery mildew. And oh shoot. Okay, there it is. But with any new adoption to a tool, the potential pros and cons, they must be addressed. So UVC has great pros. It can be used in wind and rain. We were out there in the rain. I didn't bring any rain gear on, and so I was in a trash bag, but like the unit was fine. The generator was great. Even though there was moisture on the canopy, it wasn't flicking up into the light. So, and there would be ways to really make that waterproof and better. So, and days that it was very windy, it was very safe to use. So that was fantastic to be able to not have to rely on weather, and, you could actually stick to your spray schedule. There is immediate re-entry to the field, which is great for workers, especially as re-registration for some pesticides happen. So where you do have those longer re-entry periods, and it can fit into a sustainability pest management program. It is, though it will include, tractor passes, but it is reducing your fungicide use. It's helping maybe potentially extend your synthetic fungicides that you do have, and helping rotation as we need to keep rotating all of our frat groups. And but there are some cons. So it is an increase to labor and equipment use. So we really saw the best efficacy at twice weekly. That's a lot of tractor time. There are some robotic units that are happening, that are out on market, that are available. I don't know if they are reasonable for an operation sometimes, but they are out there. And so that would help solve that issue. And it is a new management tool, so there are barriers to add this into current management practices. It would, require some research on the growers. We still have questions that need to be answered. It's sometimes hard to get some of those UVC lamps and figure out what exactly to get. There's a lot of new companies coming out that are having UVC. How do you navigate that? So there's just some barriers to entry there. And we don't know the benefit or the effects to beneficial insects in the vineyard. I do think it is low for beneficials, because beneficials aren't typically active during the day or during the nighttime, or they're a little hidden at least for UVC light. I will say that we have used it to, or we did monitor leafhopper populations while we were, and that's not a beneficial, that's something you don't want, but it did not reduce our leafhopper population as we did these treatments. So, insect control still needs to be explored with UVC, but with our with what our observations of seeing some of those pests that we were trying to potentially control, we didn't see. So maybe the effects to beneficials would not be bad. Current studies that are happening within Michelle Moyer's program at WSU. My previous program is Jesse Stevens, a master's student, is continuing the UVC work using my cool prototype. And he is looking at UVC treatments applied only during the critical window period, so that bloom period. So we found that, with our research, the critical window period, it again proved how important applying heavy hitting products are to help with cluster disease. And so he is using frequency of weekly UVC and twice weekly, and then an unsprayed control. So he is applying sulfur up until bloom, and then at bloom switching to UVC, and then ending after bloom with a synthetic product to close out the season. And then nested within those UVC treatments and the unsprayed control is canopy management treatment. So no canopy management, shoot thin plus pre-bloom leaf removal. And I just had some verbal, like a talk with Michelle, just to kind of maybe give a little bit more of an update from it, because their research is still being conducted and you'll hear more about it in the coming years. But the UVC plus canopy management isn't improving the treatments, just because they are it's not a season long. They're applying it at bloom when those clusters are visible. But they are seeing that if for the unsprayed control, those canopy management practices are very much, you're seeing the differences with disease. So no canopy management versus the pre-bloom leaf removal is dramatically different between that unsprayed control. So again, they're proving, which we know how important canopy management is. But I think it's neat that the UVC treatments applied at bloom at that critical window period are comparable to the fungicide standard. So that is really neat. So you can, target UVC at a critical window time, and it seems to be working. So that is awesome. I'd just like to take some time to acknowledge people. The Moyer Lab, our funding sources from the Washington State Wine, Grape, and Research Program, and Dr. David Dory with Cornell University, Vine Tech Equipment. They helped put together my second prototype. The ones highlighted are my co-authors for the paper that is now available to read and see. So that's neat. And with that, I'm happy to answer any questions. Again, the QR codes, the middle one goes to UVC resources, so how to build one, some parts list. The other one is the paper. I didn't include it, but I do have a paper on grape mealybug where it was kind of inconclusive work. It was laboratory work for that. That's my email address, and I'm now not at WSU. I am now at NC State, but you can reach me there. And you can also reach out to Michelle Moyer. She's happy to answer any UVC questions. And yeah, thank you so much.
All right. Thank you, Alexa. That was fantastic. We have some questions in chat, and if anybody wants to, again, address audibly, please just signal by raising your hand and we'll get to you. I'm going to try to work through these. The first question here is from Jeff. And this sort of got addressed, but he's just asking, this was sort of midway through the presentation, about parts of the cluster or the canopy architecture that don't receive light and the risks of developing mildew later in the year when the canopy is more built out and it's tougher to get all the angles.
Yeah. And so that is a concern with this, prototype, is those interior clusters. How do you get the light? Having it over the row with a VSP, and that I should have mentioned, that was the trellising that we were working with. It was a modified VSP where it was very floppy and loose. But it having the top lights kind of allowed for penetration below, and then having a reflective material that bounces the UVC light off. We're hoping to kind of be able to have it reflect and be able to penetrate. We didn't get any readings with our calibration tool to see, how much it was inside the canopy, but that is it's not like with the sprayer where you have your air and it creates a really nice swirling effect to get, underneath the leaf or, on the other side of that cluster. It's not possible. So I think that really just proves like you need to allow clusters to be seen. If you can't see them, your light probably can't see, the light can't penetrate them. And also, it's maybe like in combination, using UVC early season and then the hard hittings that, cluster are making sure you're really having that canopy early in the season. So then that's when you're using your UVC light because the light can penetrate those clusters.
Yeah, I mean, I'm looking at these questions, and there's one more sort of unique question that we'll get to, but there's an ask about GDC, like wider trellis systems. I mean, if you're using where I've always thought this would work, and I'm not trying to detract from anyone who's currently marketing this technology, but I don't think the success is in the autonomy as much as I do the timing and the volume of units. And so these things can't be that expensive to build, once you start cranking them out. And if you can knock your inoculum down when the canopy is small successively, over time, it's just like you're gonna, continue to eliminate potential disease, and this becomes a really important cornerstone in an IPM program.
Yeah.
I'm really
And I
excited.
will add to that, we were using a very small, like it was this vineyard block was a research vineyard. We had high inoculum. I know we had low disease pressure years, but that's why in 2020, I think we had such high disease pressure because that was like we had our unsprayed control right next to our UVC. So it was like constantly under pressure. And I think David Goodori has found like in larger scales where you don't have so much, pressure and your inoculum is lower in a vineyard setting where you have been controlling for powdery mildew throughout the years, it was much more successful. And it could be suitable for those different trellising. You'd have to kind of maybe design your unit specifically to that, like wherever your fruiting wire is, make sure that that's where the light is really, going. And if it's wider, maybe you have it wider and like lights different. So I think for each trellising, you'd have to kind of reconfigure the unit. And like you're saying, it is fairly affordable to build it yourself. You'd have to, kind of know what you're doing a little bit, but I was able to build it. So I don't think they're with growers who are much more ingenuity and smarter, about building all sorts and fixing tractors or sprayers. I think it's very possible to build this for your canopy architecture.
Very cool. We have another question from Molly here. She's just curious about whether or not there's efficacy of UVC on bunch rot disease. You sort of addressed that, I think, at the beginning, but
Yeah, I know. I would follow up with the pathology lab at Cornell University. I know with bunch rot and then with sour rot and I think it was mostly sour rot. They were seeing like it was inconsistent because it's a surface sterilizer, and once you have like the rot, once you have infection or the spotted wing Drosophila has punctured, the sour rot or or you have sunburn and the pathogen is in the grape, the UVC cannot cure it. But they have seen where it's basically you're using it as a disinfectant, keeping it clean, and so it allows like not pathogen. So it's looking like promising, but I don't have like a straight shot. Like, is it efficacious? They're still working on it, but it's some neat research that's coming out.
thank you very much.
Mhm.
Do we have any more questions? Anybody out there? Well, Alexa's info's up here, so please document it. And yeah, I would love to stay in touch. Personally, this is something that we continue to talk about, and how to use it, when to use it, all those things. So So I really appreciate your willingness to join us today, and I look forward to continuing to look at um, this mechanism for control.
Yeah, yeah, I think it's promising. It's definitely not the silver bullet, but it's really cool. It's working, and I think it's fantastic. I worked with some other odd machines to control powdery mildew, and they weren't as great. So it was cool to have at least one machine that was working. And I think with future research that's happening at WSU, and at Cornell, I think there'll be some good management integration as maybe growers take this technology on and do their own. I think that will also add to how do we fully integrate this into our disease management programs. So thank you.
Awesome. Well, thank you again, and have a wonderful weekend. We appreciate
Yay.
your time, and maybe we'll just take a little 5-minute break here, so people can excuse themselves and do what they need to do, and then we'll keep on rolling.
Well, thanks again, guys.
Take care. Christina, does that sound good? 5 minutes, and then we'll reconvene. Maybe 7 minutes,
That is
9:25.
fantastic. Thank you
All
so
right.
much. Let's plan to reconvene at 9:30.
Hi, Valentina.
Hello, hi.
Thank you for joining us.
Thank you for
Everyone.
inviting me. I'm honored to be part of this.
We are honored to have you and excited to hear about your research.
Thank
Um,
you.
Hopefully everybody's back from the break. Trying to see how, but yep, looks like we got everybody more or less back online. I just want to take a minute before Valentina presents to readdress the fact that we do these every year and we want the presentations to be for you as the audience. And so please don't hesitate to reach out to me or to Christina or Kristen or anybody at the Farm Bureau with subject matter for coming presentations. We make these things available for free to students from universities that have college ag programs. That's kind of a redundant statement. Universities with ag programs across the world at this point, and so the help that you give us in terms of sculpting the curriculum, it's not just for the benefit of people in Napa. It's going much further and potentially, helping to develop folks who are going to be instrumental in the cause all over the place. So thank you for that, and thank you once more to our sponsors. Barber Vineyard Management, ETS, Grow West, and Wilbur Ellis. We really appreciate your support. And then I'm going to introduce Valentina here. Valentina, um, can you pronounce your last name, Valentina? Because
Ricciardi.
Ricciardi.
Yes.
That's how I should say it. As a research fellow at the National Research Council for Wine Grapes and Agriculture Beyond that, I assume, in Palermo, Italy. She's also a fellow at the Institute of Biosciences and the National Research Council of Italy. She received both her master's degree in agriculture, biotechnology, and her PhD in agriculture, environment, and bioenergy in 2024, 2019, then 2024 subsequently at the University of Milan. During her study, she dedicated her research to the identification of grapevine genetic resources involved in biotic and abiotic stress resistance, combining molecular biology, bioinformatics, and in vitro culture techniques. Her current work is mainly focused on the identification of powdery mildew and downy mildew and breeding programs, both traditional and alternative or modern breeding techniques to combat these pathogens. Thank you very much for being here again, Valentina. We look forward to what you have to say.
Thank you very much. I'm going to share my presentation. Okay, so thank you very much for the opportunity of being part of this event. I'm really happy to be able to share with you our work. I currently represent both the University of Milan, where I used to do my PhD and my previous studies, so the first postdoc, and now I'm here in Palermo to study some more things. So today I'm going to speak about some of the novel loci we identified, loci of resistance to downy mildew and powdery mildew. Interestingly, we were able to find them in wine grapes. So we are talking about Vitis vinifera. So back to breeding as at the beginning. So breeding of resistant varieties, as we know, is one of the most promising defense strategies for fungal diseases, especially. So what's interesting about these new genetic resources is that they come from a special place in the story of domestication of grapevine, which is the cradle of domestication. And these resources can be interesting to be exploited in breeding programs. And on top of that, teach us some insights to develop other alternative approaches of defense to the diseases I'm talking to you about. Just a brief overview again. So we're talking about downy mildew and powdery mildew. Downy mildew caused by Plasmopara viticola and powdery mildew by Erisiphenecator. These diseases are particularly problematic for grapevine production because these pathogens are biotrophic and they can exploit the metabolism of grapevines to succeed in their infection. So they are able to infect all the green tissues of the plants, and this leads to huge losses both in terms of quality and quantity, as we know, of the grape produced. Usually, resistance to these diseases can be found in American and Asian species that co-evolve with the pathogen. So some examples are here in the presentation and were shown before in the presentation of Matthew Clark. And because these varieties and species co-evolve with the pathogens. This is the example for powdery mildew, and here you can see some for downy mildew. Sometimes they are shared because the loci of resistance are able to function against different pathogens, okay? Some years ago, however, researchers were able to find other sources of resistance also in wine grapes, so Vitis vinifera. This was the case of REN1 for powdery mildew, and recently in some varieties from the Caucasus, among which you can find this variety, Galoblishvili. So the first identified Vitis vinifera resistant variety to downy mildew. Why these varieties are interesting is because they retain some wild traits from the original Vitis vinifera species. And these wild traits are able to confer resistances to many stresses, among which abiotic stresses and biotic stresses. So I'm going to use the studies we did on this variety to give you some insights about how these resistant sources works, and hopefully to give you some interesting stuff to think about, to develop, okay? So Galoblishvili is special because it comes, as I said, from the cradle of domestication, and it has a unique mechanism of resistance against downy mildew. So the first thing researchers discovered was that by studying the RNA sequencing of this variety, comparing it to susceptible Vitis vinifera varieties such as Pinot Noir, the variety showed that the expression of the genes related to resistance were specular between the resistant and the susceptible variety. So genes that were highly expressed in Galoblishvili, the resistant, were highly downregulated in Pinot Noir, and vice versa. So those genes were involved in many different mechanisms, all involved mainly in structural and chemical defenses against the pathogen. So we have for example, the deposition of sugars, so like callose, that is used by the variety to encapsulate the hyphae of the pathogen and relent the progression of the disease, so to stop it in the first 6 days of the infection. Then the variety is able to produce defense-related enzymes and some other compounds in the secondary metabolism, such as volatile organic compounds, VOX. So with these insights, we try to formulate alternative strategies to fight the disease itself, such as the use of these volatile organic compounds to protect susceptible varieties such as Pinot Noir. Okay, so in this picture, you can see the expression of some genes involved in the production of the volatile compounds. And here in the pictures, you can see leaves of Pinot Noir treated with the VOX produced by Galoblishvili during the infection. And as you can see in the treated plants, wait a second, I'm going to take this down. The pathogen was not able to grow in the same way. So the infection was stunted and stopped compared to the untreated sample. Okay, so these volatile compounds were very effective in the defense of the plants against the pathogen. On top of the production of these particular metabolites and defense structure, we were able to see that some particular genes were present in the transcriptome of Galoblishvili. So usually, when we talk about resistant varieties, we hear about resistance genes as before. So resistance genes are genes that are expressed in resistant varieties, and because they are expressed, we have resistant phenotypes, okay? However, Galoblishvili allowed us to identify some other genes that are called susceptibility genes. So susceptibility genes are genes present in susceptible varieties, and when they are expressed, they lead to susceptibility to a certain disease, such as downy mildew in this case. What's interesting about these genes is that if we turn them off, like with knockout processes such as genome editing, for example, we can gain resistance to diseases and obtain resistant phenotypes. So in Galoblishvili, we were able to find at least one candidate gene of susceptibility that we were able to confirm with the novel approach of screening, which is transient RNA interference. So briefly, we applied some exogenous double-strand RNA molecule that target the gene of our interest. Okay, so Vitis vinifera BD1F7, which is a gene involved in the hormonal crosstalk in the defense pathway of the plant. Okay, the pathogen is able to hijack this gene usually and succeed with the infection in susceptible varieties. Okay, we turn it off by spraying these molecules on the plants, causing RNA interference, which is a mechanism of the plant, which is able to silence genes, okay, naturally. And the silencing of this gene lead to a strong reduction of the disease symptoms in Pinot Noir, the susceptible variety we used in the study. Okay, so by strong reduction, I mean 40% of the symptoms were gone in the treated plants with these molecules. This means that the gene is one of those susceptibility genes that is currently studied to be knocked out to obtain resistant Pinot Noir to downy mildew. Okay. So after the study of these, of these kind of genes, we moved on to the more classical ones, so resistance genes present in Galoblishvili. And to do that, we tried to identify the genetic basis of the resistance, so to identify where in the genome these genetic resources were. And we were able to identify 3 new loci of resistance to downy mildew, which were RPV29 on chromosome 14, RPV30 on chromosome 3, and RPV31 on chromosome 16. Once we identified the position of these loci in grape, we went on to study their content in terms of gene and the structure of these genes inside the genome of Galoblishvili. So in this picture, you can see a brief overview of how long the regions are inside the genome in the reference, which is a variety called PN4024, susceptible to downy mildew, and the 2 copies of Galoblishvili genome, because grape, as they already saw in the case of vinifera, is a diploid organism, okay? The first thing we can notice is that the regions tend to be bigger in Galoblishvili compared to PN4024. And this is because of the presence of multiple transposable elements and repetitive regions that are not present in susceptible varieties, okay? Another thing we can notice is that in the case of RPV30, we weren't able to define exactly the region, the position, because we need more cycles of breeding to do that. And currently people in Milan are working on it, okay? So first thing I'm going to show is the content of the first locus, RPV29 on chromosome 14. So here is shown the gene content. The arrows in the picture are the genes, okay? In PN4024, the susceptible reference variety to downy mildew, and in the 2 copies of the genome of Galoblishvili. All the genes that were found in this region were related to defense mechanism, constitutive defense mechanism. As we were saying before, they are involved in structural and chemical defenses, okay? Except this one that is more involved in the primary metabolism of the plant. So the first thing we can see is that Galoblishvili, interestingly, doesn't have more genes related to resistance than the susceptible varieties, but it's actually missing some genes. Of which we don't know the function, okay? And this could probably mean that on top of the resistance classical genes we speak usually about, Galoblishvili is resistant because it's missing some susceptibility genes probably that are actually present in the susceptible varieties. Or because it has the genes modified, those susceptibility genes, compared to the susceptible variety, such as in the case of this copy in the genome, okay? This gene is shorter, it means it cannot function in the proper way. And as you can see here, I'm going to show you some function related to these genes. We have the production of reactive oxygen species in response to pathogens, which was a gene constitutively highly expressed. We have the deposition of cuticle, which is a developmental process in plants, regular developmental process. But on top of that is a known response to biotic stresses. And finally, we have the gene that was modified, and this is probably another candidate gene of susceptibility to downy mildew, because this gene is involved in the flux of sugars in the plant, and in literature is already present material about its involvement in basal resistance mechanism in a broad range kind of manner, so against multiple pathogens. Okay. When we had the look inside this locus, we moved in the other one, which we were able to study in depth. And this was RPV31. So again, differently from the first locus, in this case, we had way more genes, okay, in kind of the same length of region. Again, we had more genes inside the susceptible variety, so PN4024, and we had less genes in the resistant one. Again, so we're talking about probably loss of susceptibility. All the genes present were involved in defense mechanism, and the genes show small differences in the sequencing if we compare the two varieties. So these small differences means that probably the genes are differently regulated in the infection. So we went to take the data about the expression of those genes, and we were able to see that actually Galoblishvili is faster in the response to the pathogen. So the response happens in the first 24 hours after the infection, while Pinot Noir responds just 3 days after the infection. And probably this late response is due to the presence of the differences in the sequences of the genes. Again, all the genes involved in defense responses, I put here some examples for you, such as serine proteases. Those genes are involved in the establishment of successful resistance, both against downy mildew and powdery mildew. There are some very interesting work in literature about these new candidate R genes. Those genes were constitutively highly expressed in the variety. This means that, again, the variety is very fast in the response against the pathogen. It's ready to face infection, okay? On top of that, we have the genes that confer the capacity to the plant to recognize virulence factors from the pathogen and activate all the metabolic pathways involved in the defense mechanisms. We have the production and the regulation of secondary metabolism and the hormonal crosstalk with the auxin signaling pathway, other hormonal crosstalk, so ethylene, jasmonate, and salicylic acid. And on top of that, we have the regulation, again, of secondary metabolism, this time in the pathway of the sugars, okay? So the callose deposition we were talking about at the beginning. So to summarize what we discovered by studying Galoblishvili is that the mechanism of wine grapes resistant to downy mildew is probably based on the loss of susceptibility factors and to the capacity of the plant to differently regulate non-host specific defense genes. And this results in the fact that Galoblishvili is more prepared to face the infection compared to Vitis vinifera susceptible variety, and is fastest in the response to the pathogen. So if we think about using these kind of resources in breeding programs, we can think about mixing these loci with stronger ones from American and Asian species that in this way we can decrease the selective pressure on the stronger loci and make resistance more durable in the field. Now, to not talk just about downy mildew, we went on to study other resources from the same place of the world, so the Caucasus, the cradle of domestication of grapevine. We screened other varieties since we found the first one, both for powdery mildew and downy mildew. Okay, and for the first thing we did was performing some phenotyping Here I describe the kind of phenotyping activity you perform on downy mildew and powdery mildew usually. It is slightly different in the material you use to perform the screening. Usually the screening for resistance to downy mildew is performed on leaf discs, while for powdery mildew is performed preferably on whole leaves. The process includes the attribution of a value of resistance or susceptibility, a score, okay? Where 1, it's when you have the whole surface of the plant material covered by the infection, while 9, the highest, is when the variety is actually resistant. We performed data collection for 3 years, and we consider data that were consistent at least in 2 years when comparing the data for the next processes of the analysis. At the end, we were able to identify 7 downy mildew resistant varieties and 25 powdery mildew resistant varieties. So very interesting data. We performed some analysis about the genetic of the population we were studying. So essentially, the population was composed of 2 kinds of Vitis vinifera varieties from the Caucasus, cultivated grapes and wild ones, okay? I think we can notice in this picture that sometimes we think that some grapes are wild or cultivated, but we discovered that genetically they are not, okay? So there are some mistake of labeling in what we think in the field is happening. But beside this small parenthesis, we were able to perform another genome-wide association study to identify some new loci of resistance, and we succeeded in that. We identified 2 new loci of resistance to downy mildew and 2 new loci of resistance to powdery mildew. So the 2 of downy mildew are on chromosome 15 and on chromosome 16, while the other 2 are on chromosome 6 and on chromosome 17. Once we identified these genes inside the genome, we went to see the content of them, again comparing susceptible variety of reference, PN40024, and the resistant vinifera we had the genome of, Galoblishvili, which is our new reference. Unfortunately, we don't have yet other genomes to study the content of these regions in them, but from the data we had available, we were able to see that, again, we're talking about regions of the genome that are full of resistance genes, but that are different in gene content, okay? So again, probably we are in the presence of loss of susceptibility in these grapes compared to the other ones. Here I show you some contents, okay? So the first category of genes we found in all the loci were resistance to pathogens related genes, okay? So in this case, we're talking about many different plant processes. So we have the regulation of cell death and defense responses in vascular tissues. This is the kind of mechanism of resistance which is for different kind of pathogens. So probably we're talking about also some vascular infecting pathogens. This mechanism is again involved in the production of reactive oxygen species that the plant produces in response to the pathogen. We have the post-transcriptional regulation of phenylpropanoids metabolism for the Kelch repeat F box protein gene. So again, this is a regulation of secondary metabolism. So it produces particular compounds that are not well liked by the pathogen, that cannot succeed properly with the infection. We have the recognition of virulence factors, such as in the case of Galoblishvili that we saw before. We also have some other kind of genes, so receptors. In this case, we were able to find receptors in at least 3 loci out of the 4 available. The most present receptors are divided in 2 kind of genes. So cysteine-rich receptors and LRR receptors kind. So they are both involved in the reception of the signaling from the pathogens. that then activates metabolic pathways of the fence through hormonal signaling. So in the first case, we're talking about the regulation of the metabolic pathway of salicylic acid that then leads to the activation of secondary metabolism responses. In this case, the molecules recognized by the pathogen can be different. So in the first case of this specific gene, we're talking about toxins. In the second one, it can be some elicitors and other molecules produced by the pathogen. Then, interestingly, we were able to find other susceptibility genes that are present in the varieties. So this could explain probably why those varieties are resistant in the first in a particular time, a range of time, let's say. Because usually, as in the case of Galoblishvili, we have resistance in the first 10 to 15 days of the infection. After that, if the environmental pressure goes on for too much, like the pathogen is too strong as a strain, usually some sporulation comes and the resistance is decreased in the varieties. The same happens in the case of these other varieties from the Caucasus region. This is probably because the resistance is not based on the recognition of a specific pathogen because they didn't co-evolve with it, but because they can recognize pathogens in general and respond to them. And the presence of, and this is because in the genome, probably, they retain also some susceptibility genes that are used by the pathogen to succeed in the infection, such as is the case of transporters. Transporters that when they have modification in the structure of the gene can lead to resistance though. We already know that. So here I show two examples of it. In literature, we have, I think it's tomato and tobacco in the other one. So they are involved in the first one in nitrate metabolic pathway assimilation. So they are related to reduce susceptibility to pathogens when they have a particular structure in resistant varieties, because they cause changes in the salicylic acid pathway again, and then they cause the regulation of secondary metabolism. And the other one is involved in the transport of different kind of molecules. And they are still linked to reduced susceptibility to pathogen, but this time they regulate the pathway of defense related to jasmonic acid. And so we're talking about constitutive defenses again. Finally, another kind of genes we were able to find in this region is the genes that are involved in the production of specific metabolites that are that have antimicrobial functions. Okay, so we're talking about the 1,4-coumarate-CoA ligase and the neomenthol dehydrogenase gene. They were characterized in different plants already. The first one is related to the activation again of jasmonic acid signaling pathway. And they lead to the deposition of lignin and the regulation of the rest of the metabolism, because lignin can cause a feedback effect that can then activate other genes that are related to defense responses. The other one, is involved in the production of neomenthol, which is in itself an antimicrobial compound. And on top of that, it's again as a feedback mechanism that positive regulates defense against multiple pathogens. In this case, it has been already characterized. So to summarize all the stuff we saw in this presentation, what Caucasian grapes teach us. So we're talking about resistant sources in wine grapes. This germplasm offers different sources of resistance to both downy mildew and powdery mildew. Now we do know that. We have already identified some of the loci present in some of these varieties. So we have RPV29, RPV30, and RPV31. In Galoblishvili, these loci could be used to develop markers and to be exploited in marker-assisted selection in traditional breeding programs. On top of that, we have found two new other loci in the population from the Caucasus, so RPV36 and RPV37 for resistant to downy mildew, REN14 and REN15 for powdery mildew. Again, these loci are currently under study to develop markers that can be used in marker-assisted selection, again, to be exploited in traditional breeding. On top of that, we can think about using these resources to perform alternative breeding solutions, such as those based on new breeding technologies like genome editing or RNA interference. So we can think about turning off susceptibility genes, knock them out, and obtain in this way precisely edited plants that are resistant to probably different kind of stresses and not just pathogens. On top of that, by studying how these loci work in the plant, we were able to get inspiration to develop alternative strategies of defense against these diseases, such the use of volatile organic compounds or the use of the technique we use to screen them, so double-strand RNA application. About that, I want to open a small parenthesis about the future steps we are working on currently. More on top of using these loci in breeding, we're thinking about using these genes, the genes of susceptibility we found, as a treatment in the field, hopefully one day, to the diseases themselves. So the plan is to use double-strand RNA to silence transiently the susceptibility genes in the susceptible variety we use to produce wine or table grapes. And by silencing this gene, we can achieve resistance as a treatment, an alternative to fungicides. While working on finding new candidate gene of susceptibility, and we have many more actually by studying these varieties, we are thinking about other approaches to make it more efficient, like the treatment more efficient, because currently the drawback is that we have to use a lot of RNA to treat the plants, and doing all this RNA is very expensive, okay, in terms of synthesis. So if we are able to make it more efficient, to make the treatment more efficient, we can actually thinking about testing this treatment in the proper way in the field. And we want to do that by using what is called extracellular vesicles. Maybe you already heard about them. Those are vesicles produced by plants or bacteria. Like, for example, we're thinking about using the ones produced starting from mast as carriers of the double-strand RNA molecules. In this way, by spraying this treatment on the plant by using airbrushes, for example, we plan to silence the genes and hopefully have a way to fight these dangerous fungi infecting them. So with that, I think I concluded. If I can go on. Okay. I want to thank some organization and people involved in the work I presented to you. Currently, I am in Palermo, so I want to thank my new supervisor, my previous group of work, which is in Milan, under the supervision of Professor Gabriella De Laurentiis, the University of California, Davis, that helped me in the bioinformatic analysis, University of Georgia that allowed us to perform this interesting research on these fantastic genetic resources, CREA, which is another research institute in Italy, and Kijin Company. On top of that, here I share some ways of contacting me, so my email and some social media. And I want to thank you all for your kind attention. And if you have any questions, I'm here to answer to them.
Thank you so much, Valentina. That was fantastic. Really interesting work that you're involved with. And I am looking at the chat to see here if we have any questions up in chat. And if there are questions audible again, please ask everybody for Valentina. Her contact information was available, so and we
Yeah,
can
I'm
provide
going
that
to share it again.
Yeah, maybe we'll have some follow-up via email
Okay.
if nothing pops up. I got a quiet group today, but thank you very much. That was again really timely and important stuff you're doing, and we appreciate you making the time to join us
Thank you.
remotely. This was great. Have a wonderful weekend, and
Thank
I
you.
look forward to staying in touch.
Thank you very much. Have a
Bye.
good weekend.
Okay, we're gonna roll directly into our next presenter, who is no stranger to any of us. He is a guy who has supported these programs for as long as I can remember. And I'm a huge fan of everybody at the Ag Commissioner's Office. We couldn't do this without the partnership that we've shared since the beginning of the program, but especially Greg, Music. He has worked for the County of Napa in the Ag Department for about 20 years, specializing in pesticide use enforcement, managing agricultural pests, and addressing pest challenges in urban and natural environments. Before that, he spent 2 decades managing turkey rearing operations in the poultry industry. Reflecting on his career, he notes, I never anticipated after graduating college that my work would revolve around combating pestilence in its many forms, from aphids and yellow jackets to viruses, bacteria, and occasionally human-related challenges. Greg Music, thank you so much for joining us.
Thank you for having me. You can hear me okay?
Yeah, we got you.
Now I will attempt the share of the screen. This will be interesting if it works. I am here. All right.
There you go. Yeah, just hit that presentation.
You guys are seeing the presentation fine, or are you seeing the
We're seeing
presentation
your
view?
screen. If you want, if you want to go down in the bottom corner there, of that last field that we were on. There you go. Yeah, we're good.
That's good. All right.
Awesome.
I may turn off my camera, or if somebody can turn off. Nobody wants to see me staring at them the whole time, but you're fine if you want to. Thank you for the nice introduction, Eric. I appreciate it. Welcome, everybody. I'm glad everybody could make it. A little cornucopia of pesticide issues. It's going to be kind of a simple presentation today, especially after the last one. I can't make out some of those words that she was saying, so I'm not smart enough for that. So they put me in charge of pesticides. Anyhow, I am going to kind of go over a few common violations that we see, and then I want to kind of address how those common violations get escalated to higher fines, higher trouble, if you say. And then I'm going to kind of approach this little thing of we had a few events this year, pesticide-related events that were done, and they were done by people that had lots of experience, and they made some mistakes. And why was that? Probably apathy. So just kind of want to address that, so everybody kind of going into next year and into this. A lot of us do training this time of year, the new year, before the spray season really gets going. So think about that when you're doing your trainings. So application specific information, I think we're all kind of familiar with this. I have the form coming up on the next slide. Pretty self-explanatory. Where are they and why do they need to be there? Are they complete? And are you training on them? Your guys should know what's going on and what they mean. So. All right, so where are they? We've seen them in tubes, we see them in mailboxes, we see them in bulletin boards. A few things on this. The tubes are great, but as we can see from these pictures, let me turn on my laser pointer here. You can see these tubes. They're fine, but not everybody can reach a tube that's at the top of a tall pole. Some of these tubes, they're too small. can't get a hand into them. This one is probably the best example. Application-specific information has on there what's been treated and when. And if you're arriving to a ranch and you want that question answered for yourself and you walk over to this tube, you could be walking right into an area that just had some pesticides applied to it and contaminate yourself. So that area might not be a good area for you. I really like these boxes. I've seen them in mailboxes, and definitely up on boards. Those are all fine applications of them, and having them there is fine. All right, let's get into the form itself. When we're out there, we're looking for complete forms. So, we want to see things like, okay, it should have wine grapes on there. Does it really need to have wine grapes? Not necessarily so. If your whole site is nothing but wine grapes, that you don't need to put it up there. But if you've got more than one commodity on your site, in Napa, we don't have a lot of that, but you could have places that have, olives, for example, or you could have some sort of vegetables growing. Make sure you identified what got treated. And then also, the second example I have here is, make sure you identify a block. You may have a big enough site that you have it broken down by blocks, and make sure you write down which block got applied to. We have had accidents where people have gone into a block that they didn't know was treated, and they checked the rest of the place and didn't have treated, but a block was treated. So make sure you fill that out. Again, start and stop times. If you go overnight and you started on one day and you finished on the next, make sure you reflect that on this thing. So definitely start and stop times. Remember, these REIs are important. Definitely this, definitely this, definitely this stuff. I put this quinoxaphene on there. That's the first ingredient in Quintec. It goes on for a long, long time. If you've got that on there, is fine. I also want to remind you, this is not the only way to display this information. And we'll get into that. Work orders, I see a lot of work orders that people write up. Either some of them are out of the CAN program, some of them are, PCA work orders, and they'll work. That does fine. Just make sure you update those things when you're done doing your application. So again, are you training your employees? Where are they? Where are they? What do they mean? When should you use them? Update that thing. So some of the common questions, if I'm doing a field worker application, and I'm going to go in there and I'm going to say, do you know what's been applied in this vineyard? And they may say, a crew may say, nope, have no clue. And I'll say, how do you know it's safe? Eric told me to go in here. It's fine. I trust Eric. That's our first mistake. But that's fine. That's a fine answer. I'm okay with that. But, they don't know what's been applied. They don't know what the rates or any of that stuff. But I'm going to ask them, do you know where to look? And that's when I want to say, I want to hear from a crew, if it's your own crew, they should be saying things like it's at the end of that row, or it's down at the front gate in the mailbox, or it's on the bulletin board over by the barn. As long as they know that, that makes me feel really confident that they've been trained. If they don't know that, then I'm not so confident in your training. So we're going to probably dig a little deeper. But if they tell us where it's at and I can go look at it, I'm pretty happy then. And then I do want to say, update those things within, the law is 24 hours after you finish an application. You've got to update those application specific forms. There's no reason why you can't update them right when you're done with the application. That's probably the best time. But also you want to tell your employees, what are these there? They tell them if it's safe to go into a particular vineyard or block or whatever, and they should understand that. They should know where those maps are of the vineyard. They should know, don't go in there, don't go here. That's fine. And when should you use them? Those application specific information has to be up after you do an application, and they have to stay up for 30 days. If you haven't done anything else, they can come down. Usually in the spray season, I'll see work orders for, or on the bulletin board, an application thing for the whole season. And that's fine. That's actually good. Remember, now also you have to prove that you had those things up. So either keep them and file them, or take a snapshot of them at the end of the season or something like that, just in case, because we could go out there if there is an incident and you got somebody that went in to get some medical care and they didn't bring the application specific information with them, you're going to be in trouble. Now, let's say you say, I have it, and okay, where is it? So make sure you keep that stuff. I think that's it for that slide. So I think we've talked about this in other classes, but the different fine levels. That Class C fines are usually a paperwork fine. They have a range because definitely different paperworks can cause different issues, but those are pretty minor things, unless you're a repeat offender. You're gonna, Class C's, most of us don't see that. The fines kick in when you have a Class B fine. So using our application specific example, if you don't have that posted or it's incomplete, right there, that's considered a Class B range. We're supposed to issue a fine. Now, if you haven't had any issues, you've been going along, you've never had this happen before, you're moving along fine, this is the first time we've caught it, it's probably going to be a letter of warning from us, even though it's Class B. And we have to tell Department of Pesticide Regulation why we only took a Class B. And we can say that, a grower hasn't had any issues, this is the first one, we're giving them a pass with a warning. Now, that may be fine, application specific, but that can escalate. So what if you didn't have the application specific and somebody went in to want to go look for it and they didn't find it? Well, now that gets that isn't us finding it on inspection a problem. That's in a real world application where it wasn't there for somebody to have it. Maybe they were feeling ill, maybe they were going to the doctors, and you didn't have that information. Again, that's real world stuff. They could not only affect their outcome medically, and so that's why it kind of compounds it a little bit more. Sorry. And then also, Class B can be repeat violations. A Class C can get relabeled as a Class B, gives us a little bit more fine activity that we can do. Same with a Class B, repeats of those can be escalated up to a Class A. One thing I want to say about Class A's, if they happen, there is no forgiveness on a Class A. You're going to get the minimum fine. Most likely you'll get the minimum fine regardless. That's where there was definitely an activity that happened that either caused harm or could have caused harm, like a drift. Not all drifts cause harm. A drift is bad. So that's an automatic fine though, if you drift. There's just no getting around that. So don't drift. But also that Class A, if you've got a safety thing, we can't give letter warnings on Class A's. So again, I kind of covered this before. Incomplete or missing can be an LOW, but You know, it can get all the way up to $3,000. Now, an event on that same thing, if you had an event related along there that compounded that original application specific, and you had this other thing, takes it to Class A, and the $700 isn't going to be the minimum because it's been compounded. You're probably going to get a heftier fine. Like I said, application specific starts at that Class B, since it's directly related to the application, and pesticides and workers can be damaged by that, by not knowing where that information is and not knowing that information. And I've talked about, if we discover it in its first time, it's not going to be as big a deal. And I talked a little bit about, you know, what is an event? Employee gets exposed, no information available when you take them to medical care, or incomplete information on an application specific. So they went and looked at the thing. It was incomplete, and then they walked in and got contaminated because they didn't know that that field had been treated. That's going to be a Class A violation right there. It got compounded because the application specific was wrong. So, and then also employees that are denied access to that application specific, that can result in a fine. Application specific information is supposed to be available to the employees unimpeded. So that's why we get into, you know, where you put those in the, sorry about that, I've got to pop up here. That's where application specific has to be just unimpeded access to it. It goes back to the days when foremen on ranches could do whatever they wanted, and information was value to them. So they held on to it quite tightly. And if you wanted information, you know, you had to go through them. That is not the case with pesticides. If they want to know information about what's been applied in your site, they have access to it, unimpeded access. So what's that mean? Posting it and all that. What if you're a farm labor contractor? What's that mean to that guy? Farm labor contractors, they show up to a whole bunch of different sites. They don't necessarily know where they are. Then it's on the grower to make sure that they get that before they arrive. Or to make sure they know where to look when they arrive. Hey, it's posted on the barn, train your employees so when they know, hey, it's posted on his barn over there. If we want to go look, I went and looked, it's safe to go in. Again, that's fine if we come across employees. They may not know, even on a farm labor crew, but if they know where to look. And where to look for them might be maybe not where the grower keeps it, but he gives a copy, and you know, it's at the sandwich board that they have to post for all their safety stuff, or it's on the tailgate of the foreman's truck or something, where it's unimpeded access. They have to be able to get to it. So the farm labor one gets a little interesting because, you know, you could get guys that show up that don't know where things are posted in your vineyard. So I don't like football. All right. How about missing PPE? You know, that's going to be, you know, if we discover that, I've got the commissioner here back in the days when she actually, I'll make the joke, work for a living. She modeled for us here. You'll notice the safety glasses on her head, the safety gloves in her hands. They aren't on her hands, they're in her hands. So if we come across this, you know, we see people with the safety glasses up all the time. You know, it depends on the application, but usually we're going to mark you down for a violation for that. That's a piece of safety equipment that we want to see used. Now, maybe I mark you down for that. And you're like, okay, it's the first time in a few years. It might be a few warnings, but how can that get escalated under this thing? That's a Class B action right there. But let's say if I, you know, okay, we're talking to them about this, and you know, they don't have other stuff. So let me pop into that. So again, let's get on there. We review the fine levels. So how can a missing eye protection get compounded really easy? So not wearing eye protection. And then, oh, I looked at what they're applying. It requires eye protection. Consequently, that means it requires a pint of emergency eye wash. And that doesn't matter if they're applying with a horse sprayer or an orchard sprayer. They have to have that pint of emergency eye wash. So they might be having to carry it with them. If they're using like a backpack sprayer, they got to carry it with them. Or it might just mean it's on the tractor or the ATV and it's there. We're going to see that. But that's going to compound because you weren't wearing eye protection and you didn't have this other thing. So if you did get something in your eye, it just got compounded. So as an inspector, I'm going to compound that find as well. And again, let's say you had the pint of emergency eye wash, but that got back to the mixed load site. There was no continuous eye wash available at the mixed load site. It's got to be there. Again, if you had an emergency and you don't have that stuff there, that's going to compound what's going. So it, somebody with their glasses up on their head, we give them a nod, we kind of yell at them when we're doing the inspection. We mark it down, we tell you, okay, we'll do better the next time. There is no next time. You're going right to a fine if there's these other things compounded. So, because to us, that goes back to, okay, are you training? Because they didn't set that up beforehand. So that's where we kind of get, especially safety stuff, better be there. Let's get into some of the common C violations that we see, pesticide use reports either not coming in, incomplete, or late. Usually it's a letter of warning, but if it keeps on happening, we can fine for that. School site notifications. Again, if they're missing or incomplete, so far on the school sites, we haven't really pushed those on people. We're still doing a lot of hand holding on that because it's not the best program. But at some point, we're going to start finding people on those things. Beekeeper notifications. Make sure you're checking that. We can look at that stuff. Did you check to see if a beekeeper needed to be notified? And we're usually going to check that after we find a bee kill. So, if we find a bee kill and the beekeeper got notified by lots of growers, but not you, and you're the only one that sprayed product that was toxic to bees, you're going to get, again, that common C violation is now going to get elevated because maybe you didn't follow the procedures for spraying products that are toxic to bees. All right, so I kind of talked about this at the beginning. We've had some bad applications this year, and that caused some problems. So, and they were all done by people that had experience. So I say apathy equals trouble. I kind of list off all the things that can be trouble, exposure to applicators or field workers, drift issues, bee kills, contaminated soil and water because maybe you spilled or something or didn't do it right, mix and load too close to a well, that stuff. Non-target exposure, usually that's drift. When offsite, neighbor issues, resistance issues. If you're really doing a bad application, maybe you could run into some resistance issues. I highlight the bad applications because this is what I think really pushes it home for a lot of people is bad applications usually means one of those things. Your grapes aren't going to be as good a quality. You're going to have less production. You're going to have higher cost because, bad applications can also mean trouble, where you have somebody get exposed or drifted on. So your costs are going to go up. Bottom line, bad applications means you're wasting money. So don't be a bad applicator. It's not good for business, and it's not good for safety or neighbors or anything like that. So do you have a plan on fighting apathy? Pesticide training is part of that thing. Make sure you do it well. And, some of these are, the HR idioms, to ensure good things, but there is some truth in that, foster ownership. If you've got an applicator that, hey, this is my site, I take good care of it, I want it to reflect in that, reflect his hard work. He takes care of it. Somebody that is, contributing to your team, with, coming up with, potential safety things or stuff like that. Recognize that. That's what you pay them to do. You want them to do it, so recognize it. Too many times, I don't think we recognize the common work that they're all supposed to do and rewarded for. Again, invest in training. If somebody knows that, what they're doing, it usually increases all these other things. So again, build a team, encourage support from all. In my previous job, I like to use our safety training that we did. And one of it was, see something, say something. It's not, you're not stabbing a fellow employee in the back if they're not wearing their respirator. Tell them, you're not wearing your respirator. Put it on, because if you get injured, that's going to cost you money, and which could reflect on your bottom line. And again, we talked about bad applications. If you're increasing your costs and you have less of something because you did a bad fungicide application, all that piles on itself. So that if you've got employees that are kind of watching out for each other, that's a good thing in my book. So if you need to revitalize your training, do that. Change it up. If you think people are getting kind of stagnant, change it up. Address issues promptly. If you've got somebody that says, I always use the respirator example. You got some guy that says, my respirator just doesn't fit. It's always coming loose. I want to change respirators. Then do it. Don't wait, let them wear it a few more times and he ends up not using it or properly, gets contaminated. Address those issues quickly. And that's kind of leading by example, do what needs to be done and make sure you fight that. And then I have a bunch of the idioms to fight apathy. All these things, they're cute and but they're based in reality. Lately, I think I like to take off the blinders one better, because we see a lot of people, you get guys that will go out, they're going to do an application, they've got their work order, they've got their stuff, they may have all their safety stuff just fine, but then, they get on their tractor, they fill it up and they take off. And they didn't see the house that was having a party, and they just drove by and all the people outside go running inside because they're afraid. And now you've got pissed off neighbors. So again, encourage that, when you arrive to a site, what's going on there? I've talked about this stuff before because it's something that I think the application doesn't start when the guy hops on the tractor and starts to apply. That's when the application actually started, with training and all that stuff. So that's one of the last things you do when you're applying is actual application is one of the last things. So again, I get into this, what's happening around your site. Identify those problems by site. Don't leave it up. If you've got an applicator that, teach him, look for problems that could be an issue. I talk about drainage issues. It's not necessarily, getting stuck in the mud like the next one. It could be you have a little seasonal drainage area that splits your vineyard. You can't have vineyard there because it's a seasonal creek. Well, you don't want to have somebody spraying herbicides into there either. So make sure they know about those things, where they could be problems. Service people, we get into that all the time. Are they trained? If you've got a mechanic that comes out and after a tractor broke or a spray rig broke and he's working on it and it's still got material in it, he needs to be trained. He needs to know everything your applicator knows, as far as safety stuff goes. And then only trained employees can work in treated fields. So a lot of people get confused on this. So let's restricted entry interval, REI. Every product has that. It's at the end of that REI that is now a treated field. Before it's a treated field, but it's under that application. So to enter it before that reentry interval, basically you have to be an applicator to enter that. And you should be trained as an applicator because you got to put on all the PPE that's required if you're going in there before. But if after that, then it just becomes a normal treated field. You have to be trained before you go into those treated fields, unless 30 days have passed. So I'll back up. Let's say it's the new year. You've got a few new employees on the crew. You're like, okay, we're gonna, get out there and start pruning. If that field hasn't been treated for 30 days, fire them in there, put them to work. That's great. But if it was treated in the last 30 days, herbicides count as treatment, was treated, they have to have that field worker training before they can go into that treated field. So when we do inspections, we ask, have you been trained? And, some people, we get no answers. And most of the time it's something along this line of they're a new employee. They just came on board. Maybe they've had field worker training in the past, but you don't know that. So make sure you train. Because that's one of those issues that if you don't train, they get damaged. That's an event now if they got sick or something, where before it was just us coming across somebody not being trained. That's one thing, but it escalates if they get sick, obviously. So know before you apply. Do you know what you're doing? I know that sounds a little harsh, especially, if I asked Eric that, I think he might be a little put off. Maybe not today, but yes, everybody, you're all here, you're all getting continuing ed because you know what you're doing. But that apathy works against you sometimes. I think in every one of our events that we've had this year, they've been done by people that knew what they were doing. Apathy set in, laziness set in, and then an application went south. And so combat that, think about that when you're doing your trainings, think about it when you're doing your applications. Don't let apathy set in. So are you using the appropriate chemical? Always, like I said, the application doesn't start when you start to apply. The application starts when you start planning on it. So is the appropriate chemistry being used? That happens quite a bit. Are you using the appropriate chemistry? Is the equipment correct for your application? I hit on this a little bit earlier too, and I'll probably hit on it down the road again. Is it working properly? It's not only is it correct, but is it working properly? I have seen people applying herbicides with an orchard sprayer that just had the last nozzle on down lower. I'm like, okay, no. And then what about notifications? We talked about that a lot, but I always harp on it. Schools, bees, neighbors, have you done all that? Not that you have to do neighbors, but I think a lot of us do, or a lot of us do some outreach to neighbors when it's appropriate. So is the weather appropriate? This isn't well ahead of time, but ahead of the application, you may be looking ahead. Those are all appropriate. You don't want to spray something, have it get all washed off. Again, bad applications means you're spending more money. You might not have as good a crop, or just maybe wasting money, and you don't want to do that. And then I have this last one. Any community activities scheduled. Might not be a bad idea to open up those community calendars and see what's going on. I'll use the example, as many of you know, Andre, him and I were out earlier on in his career here, and we were going up Highway 29. There was a foot race happening coming out of Charles Krug. Highway patrolmen were around. They had half the, part of the road coned off. Slow down signs, all that. And they were walking from Charles Krug up to Calistoga. And so it was pretty good distance that these people were going. And we happened to be ahead of the crew a little bit. We were up there pretty early. So the walkers weren't there yet, but we saw a crew setting up to do an application on the west side of 29. They're on the east side, probably at the edge of the vineyard, probably 100 feet away from where the walkers were. I don't know if I'd do an application then, even if all the conditions were fine. I wasn't going to be drifting over to them. I can do it fairly safely. But as we know, there's a lot of people that don't like pesticides to the extent that, they see pesticides and they may have a reaction, not because they're getting pesticides on them, but, they just think they are. So in that case, plan for those things. Maybe I don't want to be applying when they're there. If you know ahead of time. In this particular instance, we kind of talked to the crew. They called their boss and they said, and then, no, that's a good idea. We probably shouldn't do the application. And just as they were loading up to leave, they actually had a truck that was going along filming the racers and all that. And I was just thinking, can you imagine if the truck filmed a bunch of racers that were coughing and all that, and then they zoomed in on the applicator doing an application? That'd be really bad. So again, a little search of community calendars might not be a bad thing. You folks within school sites are already aware of this. If you're some applications you have to do outside school hours, but there are outside school hours, there are activities that go on that, the school is supposed to be shut down during those times, but we know there's football games, there's events that happen all the time. Knowing that, scheduling around it might not be a bad idea for you. If you don't have to do it, don't do it. Again, you can do it at night though too. So there are things even for those school people too. Am I supposed to break in the song? I don't like it. Nobody's here to look at. Let's see, next slide. Is your equipment ready? Not leaking? That's an obvious one. Sprayer shuts off and on properly. I bring that up because we did have an inspector that got sprayed as the tractor drove back to the mixed load area. It was still fully on because a valve wasn't working right to shut it off. So make sure that stuff works like it's supposed to. Is it calibrated? Take the time to calibrate your equipment. I think it was last year I was driving around, came upon saw a bunch of people out there filling up spray rigs. And I was like, yes, I've got an inspection. Then when I arrived, I could see they weren't filling them up. They were filling them up, but they were also actually firing them up and then measuring the volume of water that was coming out. And I'm like, dang it, they're just calibrating, which is a good thing for you guys. It's not good for the inspectors because it's not an application. So I was really pleased to see them calibrating them. That was good. Tractor ATV working. You don't want somebody breaking down the middle of a block and then they got to walk out through a treated area. Especially if you are I always use this example on a tractor, an enclosed cab. They aren't respirators. Enclosed cabs aren't respirators in our area. We don't use those. But let's say if you're applying a product that requires safety glasses, you may not have those on in the cab. You're behind glass. You don't really need safety glasses on in that case. You've got a whole thing there. But what if that tractor breaks down and you get out? You better have safety glasses inside that tractor. That's not an exemption. If you get out, you're now in a application area. So you have to have all the PPE required. And then service containers, are they labeled? I kind of get on that quite a bit. I use this example. We're not only talking the small service containers that you might use, just around your place to transport bulk chemical maybe, but like in this case, they've got mixed and load product in that tank on the truck. And then they drive around at night and they hit all their smaller vineyards with backpack sprayers. So they're driving around with a bunch of product. When you're driving around in your rig, it better have, if you've got, even if you're hauling a tractor down the road, pulling an orchard sprayer from one site to another, it better have what's in that thing if you're taking it to apply. So, the name of the product, the signal word, and then your company name. Obviously, you have to have labels on hand whenever you're applying. So if you're going to another vineyard and you're mixing and loading at one and you're going somewhere else, those labels have to be on hand. So make sure you've got all that stuff. Is your safety and decontamination equipment ready? Minimum 3 gallons per handler or plumbed. So you got maximum water there. That's just water, to rinse off, wash hands, do whatever. That is not your emergency eye wash. That pint or the continuous stuff, that's either a hose or those portable systems. This is just water per handler. So make sure you have that. Make sure you have all your single-use towels, your coveralls, your water, your soap. If you don't have any of that stuff, you don't start the application. All right. And too many times we come across this stuff and, we dig around. There's no water. There's no soap. We'll shut down your application. You don't want that. That's going to be a fine if we see that. So don't start and train your employees. If they don't have this stuff, don't start the application because, if they start, that's when we're going to show up. That's the way of the world. Murphy's Law, all those things. So don't do it. Again, is your eye wash ready? Do you have a hose, a hard plumb system? If you have a hose, please make sure it's clean and it's just there for either washing your hands or, washing off. It can be used for washing off equipment and eye wash, but remember that water's got to be potable. If you look up the regulations, it can't cause injury or harm if you ingest it, put it on your skin, whatever. That's the definition of potable water. So it's got to be potable. So if you're, I know we have a lot of mixed load areas that run right off a reservoir. That water in the reservoir may not be potable. So make sure that you've got potable water for eye wash. Right now, these portable eye wash stations, I know there was a move a few years ago from DPR to make it a requirement that you either had to have some sort of hard plumb system in or a portable system. I don't think that's gone through yet. So a hose is still perfectly fine. But please, if I come across a hose that I can tell has been inside a tank because it's all grody on one end or full of dirt or something like that, I'm not going to count that as an iWatch station that's ready to go. So you can argue that with me, but I'm not going to count that because I don't think, you shouldn't be applying with something that's going to possibly cause injury to your eye. Emergency eye wash again. Do you have the pint? 1 pint of water on your person if you're doing a backpack spray or an ATV. It can also be, just sitting in a tray, or we do see people that use, the bottled drinking water, which is potable water, and that's fine. But we've come across those where they've been drunk. Not the people, but the water has been drunk. So make sure that doesn't in case, right? Emergency eye wash on there with a Sharpie if you need to, but make sure that that is available, ready to be used if it's needed. If they need drinking water, have drinking water available. All right, how about it's windy, rainy? We talked about that a little bit. There are some best management practices that are some on the labels, some just kind of in general. But you may have some for your particular sites too. So do this site evaluation. I always kind of use this application. If the wind's blowing that direction, we don't apply because it's going to go into the houses in the little picture here. Or anemometers, real cheap now. I think you can get them for 25, 30 bucks. You get a couple of those, or even if it's one per kit, it's not going to break the bank. And it's super easy, especially if you're a site like the one in the picture here, where you've got urban influence over here that could really, if you can sit there and, if they start complaining and you've got, hey, every time I mixed load last night, I recorded the airspeed and it never, got up here. You can look. That's reassuring to people. And knowing that happens, I think makes people play nicer too. Is what I mean to say. But definitely know your environment around your vineyard. Neighbors can be really problematic. I think most of our neighbors understand the right to farm, so it's not too bad. If they don't like it, they know that their rights only go so far, to their fence line, basically. So, you're allowed to do what you need to do on your property, but if it leaves your property, then it's going to be bad. So, the other thing, know your site. We talked about this a little bit. If you've got neighbors over here and no neighbors on this side, just your house and stuff, maybe you start over here. If you're starting at 8 o'clock, 9 o'clock at night, this might be the better place to start. Finish up over here. The winds might be less, but also they might be asleep and in bed and not really too worried about you then. To where, like, if you're applying over here, it's 8 o'clock and they're out here enjoying their patio or something like that, they could be annoyed with you. So think about that kind of stuff. Have a plan for each of your sites. Understand what could be happening around your sites that can save you some trouble down the road. So. Talked about this too. I'm hitting on what's next door, what's near your site. We had an example many years ago of somebody doing a post-harvest VMB treatment with a nasty product, and it was late. It was down Carneros, and they were like, "Oh, we can do it. Everybody's applied." The neighbor had left some fruit on the vines, but they were like, "Oh, he just couldn't sell them. He just leave them on there." They didn't ask the question. If they would ask the question, the neighbor would have said, "Oh, I'm doing a special project for just getting a few tons for a guy that wants to make some late harvest wine." So That's why they were still on there. Now he couldn't make that wine. So again, make sure you apply where you're supposed to apply. Don't let it drift. What else? Field workers, do you have a plan? I would make this your applicators, especially those ones that are applying early in the morning, when, after sunlight comes up, after the sunrise, they could have a crew show up. And, in this example, you could have one grower over here and a different grower over here, and they share the avenue. This guy, if he sees that, stop the application. This is a big vineyard in this picture here. He may be just going away from us, and if they all moved up to this side, they'd be fine. But again, do you have a plan for that? Do you train on that stuff? You may not want your applicator making those decisions, but you do want him to call and tell you, hey, I got a field workers that just showed up. What do you want me to do? Stop. Should I, whatever, but have that plan in place ahead of time. Kind of go over these scenarios, because a scenario usually means stop, take a minute, because this is outside the normal. So think about what you're doing when it's outside the normal. For our winery folks, what's going on in your winery? Do you have other cellar workers nearby? Are there other activities going on? Are you inside or outside? Remember, sulfur dioxide, if you're applying it inside, you need to wear a respirator. If you're outside, you don't. So, not that you can't apply inside, but you better have respirators. That better be part of your training. Again, plan ahead. You can have visitors walking through a winery, that maybe you might be doing some sulfur nearby. So again, plan on these things, know what could happen, think about them ahead of time. It doesn't take that much time to do that, but you just don't. And if an applicator looks up and sees this, maybe I should stop. So sensitive sites nearby, daycare facilities, nursing homes, you could have some sensitive sites that you don't know about, and we're not going to get you for that, because the ones that you don't know about usually only show up if you're doing a nastier chemical, and they may talk about, do you have any of these sites? And those are usually restricted materials, and if you're doing that, we're going to have access to some of those other sites, because they do have other things. On some labels, you'll see hard to evacuate sites. Schools, daycares, nursing homes, those are all very hard. Some of your nursing homes may not be identified to you all the time. So that's why, on the nastier products, they have that in place. So if you plan on using a nasty product, start thinking about that plan. The plan doesn't start even when you necessarily come to the ag department to get a restricted material permit for it. You may be thinking about that ahead of time, because, geez, I got all these hoops to jump through. Let's see if I can mitigate my problem another way. So, application paperwork, is there a work order or a PCA recommendation to follow? That applicator needs to have that. You don't want them kind of, oh yeah, this is what I did last week. We've had instances of people following the wrong work orders. So be careful there. Don't sit there and, I know there was one well-publicized one where somebody put herbicide in a fungicide mix. Again, you don't want that. So, kind of plan. Do you have the right work orders to follow, right recommendations? A8s, A9s, are they displayed and filled out properly? Again, it doesn't take a lot of work to fill those out and post them. It's kind of busy work, and everybody gets annoyed with it, but they have to be there. So, emergency medical care. That isn't just 911 posted. That has to be telephone number, name, and address of where that emergency medical care is. If you don't know, ask us. We've got little labels. We'll give you some. You can slap them on things. You know, many times when we're out doing inspections, I'll come across emergency medical numbers that are kind of faded. You know, it's got it all filled out right. They're faded. We'll take one of our labels and slap it back on there just to be sure that they're fine. That is a question we always ask. Do you know where to get emergency medical care? Do you know that? And you know, the answer we're looking for there is yes, and demonstrate it to us. You know, they're going to say, oh yeah, I go to the Queen, and you know, it's there on Trancas Street. I know where it's at. You know, they may not have the number, but they know, they have knowledge of the emergency medical care. That's good. It does need to be posted though, but they should also have knowledge of it. And the knowledge doesn't necessarily mean that they know where to go, but they know where to look at your site. Oh, I know the emergency medical numbers are on that bulletin board or posted on the side of the barn over there. Emergency medical posted, not displayed. I think we've had some trainings. I think you guys had one earlier in the series about the difference between posted and displayed. Posted is a hard up, everybody can see it. Displayed may mean it's up, everybody can see it, but it can also mean, oh, it's in the binder over there. It's in the mailbox. You know, you can go get it in the tube. Emergency medical care has to be posted. That's more of a hard thing that you have to do. Contact plans, you know, are you guys, you know, hey, if you run into a problem, you call blank, blank, blank. You know, do you have those things set up? And that should be part of your training too. How about personal protective equipment? We kind of got, do you have all the stuff that's needed from the label? Look at the label. There's regulations. You know, the regulation to start with for everybody is gloves and glasses. That's the minimum that you have to do. But then read that label. Make sure you know what the label needs. Do you have all the PPE? Because some of the companies out there, some of you guys require people to wear certain PPE with everything that they do, not just by the company. So the one that comes to mind that everybody is, more and more people are doing, are safety vests. I don't concern myself with safety vests. I think of pesticide related PPE. So for me, that is, let's say a respirator, not necessarily one that's needed by us. I'm kind of separating it off. We don't require it because the chemical doesn't. But some companies, they want everybody wearing at least a dust mist respirator to start. So, you know, you have to make sure that you've got people trained up for those. If it's a requirement, they have to do you have to do the medical evaluations and the fit testing and all that. If it's voluntary, you have to have that voluntary posting up. And I will go into this, or your inspectors will go into it when you renew, or if you've got a question, call our office. We're happy to break all this down if you've got further questions too. Emergency eye wash, I kind of went over that. If the label requires eye protection, make sure you got that 1 pint of emergency eye wash in place. I hit this fully stocked mixed load site. You know, if you don't have it, don't start. Applying more than a quarter of a mile away, you better have another decon site out closer to where you're applying. We have some pretty good sized sites. You know, you can mix and load in one area and then drive through the vineyard, and it's not a quarter of a mile as the bird flies. You know, if you have to go around blocks and get there, that quarter of a mile disappears pretty quickly. So make sure you've got that stuff going. Application equipment, again, I hit on this kind of doubly. Make sure it's working properly. Is it working? Did you test it? Did you calibrate it? Make sure, even in training, again, Andre and I were out doing weekend inspections one time, and we came across a guy, and he was applying in a vineyard that had grass that was about 4 feet tall, no vines, but all the trellis system. And he's spraying and he's wearing no PPE. So we're like, what is he doing? And so we stopped him, said, what are you spraying? He had it down. I'm only spraying water because I'm not fully, prepared to do applications yet. They're training me. So they gave me a tank full of water and practice going through an empty vineyard. Hope I don't take out all the other stuff, but he wasn't. He was doing a good job. But it tickled me to see somebody getting trained like that. Driving the tractor is part of doing a safe application too. You'd hate to see somebody take out a bunch of vines, because usually if you take out vines, you're harming equipment too. And then it doesn't work properly and you can have an accident. You can have an issue. So training is a good thing. Again, proper PPE for mixed load now. So, some of the chemicals require a face shield. Not when you're applying, but when you're mixing and loading. Same, they might require an apron when you're mixing and loading. Review those labels, because it may, you may have, this is one that I think gets people too, like especially the folks that use Agrian and some of those programs. A lot of those might be defined by application PPE, not mixed load. And they'll define that in the label, but sometimes in those canned things where it's just taking stuff out, they may not get that. So be aware of that. And do you have an air gap for your spray rig? Again, I don't want to see a hose going into a spray rig. I do not want to see that. How about measuring equipment? Do you have things to measure properly? You don't want to over apply, and you might not want to under apply depending on what your pest is. So, do you have the right equipment to make sure you're doing a good application? Is there a well nearby that you got to be aware of? Remember, around a mixed load site, if you've got a well within 100 feet, it's got to be bermed unless it's uphill. It doesn't have to be bermed. If it's downhill, if it's within 100 feet, it might have to be bermed to get water around it. And that's even a concrete block. You have to have a berm around it because water can get in and go underneath the concrete slab and then down that shaft into your water. So protect those wells too. And then if you do all that, you're ready to begin the application, and hopefully these people won't be too mean to you when they come up across you. That's all. That's it. I'm done. Put a fork in me.
Great work, Greg, as always. Thank you so much. And we look forward to seeing you again next week.
Yes, I'll be back next week, but in person.
Yeah, in person. So
Oh.
next week's, we should address that now. If you haven't signed up and you're on this call, please sign up next week. It's going to be great. The subject that we're going to address is something that's important to all of us, I think, and that's ag-urban interface. We have not touched on that for a long time. It's always kind of, underlying in the different things that the county presents, but we're going to be addressing specific issues, and we sort of changed the subject matter around a lot, to make it more consistent and useful, I think, than what it looked like originally. And so revisit it on the website. The material's updated, and you'll get at least one notification via email before next week. But yeah, Greg's gonna be back, and we're gonna have a rep from DPR and some good folks.
I think you have Jen too.
The
So
reps
you'll have more ag department people to board
two
you to
ag
death,
department
too.
people, and then two chemical reps, or not chemical reps, but entomologist. And then we're gonna have a surfactants overview too, which is something I have not, I don't think we've ever done with this group, with the intention of reducing drift. So it'll be good. Please join us. Thanks a lot, Greg. I see Melanie or Milani. How do I pronounce your name effectively?
Melanie is fine. Good morning, everyone.
Melanie, thank you for joining us so much. Melanie and John had to get up very early this morning. They're in Auckland, and we really, really appreciate you guys on your Saturday getting up early to join us. The research that you guys conducted, I came across online, and I found it to be really fascinating, and it was going to be a perfect fit for this event subject that we are in the midst of today, and we appreciate you guys kind of being the cherry on top of our block of presentations. I'm going to give you. I don't have, so I'm going to introduce Melanie, and then John. I don't have your bio. If you don't mind, just saying a couple of things about yourself to introduce yourself, I would greatly appreciate that. If you feel like it. If you don't, you don't have to. Your call. Professor Melanie Kaw works at the University of Auckland, New Zealand, where she carries out research and teaching activities within the environmental science program. She studied soil science and agronomy at the University of Lorraine in France, and before subsequently developing expertise in the exposure assessment of contaminants in the UK at the University of York. And the University of Vienna in Austria, and then through the CIRO program in Australia. Her work on nanopesticides is a good illustration of the multidisciplinary and intersectoral approaches applied in her research. Dr. Kaw will present with John Habito. John, hopefully I'm pronouncing your name right, who has been conducting research on the applications of nanotechnology for wine growing since 2021. Thank you guys for being here.
No, thank you very much for the invitation. We're really delighted to be able to share the research that we do. I think this is probably going to be a bit of a change of a gear, but we hope you will like what we've prepared for you. So John is going to share his screen if he's able to. He's going to be the slide master. And I've been working on the applications of nanotechnology for more than 10 years now. And so I wanted to give you a bit of an overview of what has been going on in that field. I'm a scientist. I'm based in a university, and so I do research, but I'm trying to do research that is impactful on people who may need it. And this is what John's project was really about. It was funded by the New Zealand wine growers, looking at how nanotechnology may help wine growing. And so this is why we will co-present with John, me giving some more general fact and a little bit of context, and John will be able to dive into more specific things. So I will just need to ask John every time we need to change the slide. So John, can you change the slide, please?
All right, so there we go.
This is our outline for today. So the first part of this presentation will really give you an overview of what nanotechnology can do for agriculture and what nanoagrochemicals in particular have been demonstrated to do so far. And then John will tell you about a literature review that he conducted to figure out what may be the most promising for wine growing. And then I will take the mic back and tell you about some of the concern some people have about the potential risks that those novel nanochemicals may represent for the environment. And this is exactly what we do in our laboratory. We're trying to evaluate the benefits and risks of those novel chemicals. And we will change again the mic to John, who will share with you the work that he conducted on nanosulfur, which includes some laboratory work, but also some field work that he conducted in the USA. So if you're ready now, I'm going to start with a big picture. John, if you can change the slide, please. And make us think about what has happened in agriculture over the last century. We saw a lot of new methods being introduced. This is what people very often call the Green Revolution. So we can think about the introduction of nitrogen fertilizers, mechanization, irrigation, the introduction of plant protection products, new varieties, that all those contributed to an increase in the productivity, but also some pressure on the environment. So this is the first curve at the at the top of this graph that we see the environmental impact of agriculture has been increasing over the last century. The next curve underneath is the ratio between the yield and demands. So it basically shows that despite growing in productivity, because the population has been growing, the overall ratio has been growing, but now it's kind of plateauing and even decreasing, where there are lots of regions in the world that are facing food insecurity. And the glass curve, I'm just talking about the black part for the moment. The black curve at the bottom looks at the resilience of our agrosystems. There are a number of lines of evidence demonstrating that our monoculture are becoming less and less resilient to the changes that we're facing. Think about drought or flood events driven by climate change. And so those three curves indicates that we may be reaching the end of the green revolution, transitioning to what some people call agri-tech revolution that relies on novel technology, information technology, new varieties, new ways of transforming varieties. If you can click one more time, John. And here the objective is not only to produce more, but it's also to produce more efficiently, more sustainably, and also to have agrosystems that are resilient. And so what tools are we going to use for this agri-tech revolution? Information technology, for instance, you can think about gaining information from satellite for precision agriculture, for instance. The second point covers things that may be helping developing new varieties with gene editing methods. And the last point is about novel materials. And this is where nanotechnology stands. It's all about designing material and chemicals that are taking the advantages of what happens at the nanoscale, which I'm going to tell you about in the next slide. John, if you can go to the next slide. Yes. So what is nanotechnology? Some of you may have heard about this word and have the feeling that it's about small, very small objects. And you would be right. So this is this branch of science and engineering that looks at designing, producing, using the structures at the nanoscale. And I must say that we've been doing it without knowing it for many decades, but now our ability to watch things, to design things at the very, very tiny scale gives us a lot more power to design more efficient agrochemicals. And so to give you an idea on how small nanometer is, I must admit that I've been working in this field for about 15 years now, and it's still mind-blowing how small that is. So I'm taking this illustration today. What you see on the picture is human hair under the microscope, and you can see that the width of this hair is 100 micrometer, and that's 100,000 times larger than nanometer. So we're really talking about very, very tiny things, just slightly larger than molecules, basically. And so there's been some work by nanotechnologists to design new agrochemicals using these properties that happen at the very, very small scale. Yeah, you can go to the next slide, John. And on this slide, I want to tell you a little bit more in details what nanoagrochemicals are made of. So we may be talking about nanofertilizers, nanopesticides, so that could be insecticide, fungicide, herbicide. Here I'm staying on the concepts of what do they look like and how different they may be to the classical agrochemicals. So on the top left, this is what we call very often first generation nanoagrochemicals. These are made of nanoparticles that have been considered for other fields. So we would have carbon nanotubes, metal oxides or copper oxides, for instance. The bulky ball that some of you may have heard of, this is a tiny little carbon ball. And I would say that the things that John will tell you more about, the sulfur, will probably get into this generation where we basically only have one or two elements making a nanoparticle. A lot of my work has also focused on what we call second generation nanoagrochemicals. This is where we have a nanocarrier made of a range of materials, could be carbon-based, could be silica, Could be virus capside that are being emptied to be loaded with a substance. And a lot of them are looking at reformulating pesticides that are already on the market. So I don't know, it could be glyphosate, for instance, that you load onto nano capsule so that it can be delivered at the right time and at the right place. So it's a lot about getting more precise. There are also a lot of discussions to use those nano carriers to allow new substances to be used. Pheromones, for instance, or RNAi are type of novel agrochemicals that are being discussed, but they cannot be applied just like this. They need to be delivered. And nanotechnology gives us a lot of options to deliver them at the right place and at the right moment. So John, if you can go to the next slide. On this slide, I'm trying to summarize what the key objectives of nanoagrochemicals have been so far. And let's keep in mind that a lot of the work is still at the development stage, where researchers like us are in their laboratory designing a new nano fertilizer or nano herbicide. And we may have some goals in mind. And most of the time, those goals are to address the weaknesses of current nanoagrochemicals. And so I divide this into 3 areas here. One of the weaknesses of our current agrochemicals is related to its applications. And so nanoagrochemicals, nanoformulation may aim at stabilizing suspension. And there is a very nice series of nanoformulation on the Canadian market. And I think they are being registered in the US that really allows tank mixes, for instance, that are theoretically incompatible, but with the use of nanotechnology, farmers can mix together some fertilizers and some pesticides, and the suspension remains stable throughout the application, which has a lot of advantages for the farmers. Nano applications may also help reaching a homogeneous coverage of the crop or achieving more precise target area. The blue area looks at reducing the losses of fertilizers or pesticides that are unwanted. So think about all these pesticides that may be running off to a nearby creek, or these pesticides that may volatilize or leach to the groundwater. These are loss, which is not good for like economically for the users, but it's not good for the environment neither, because this gives a chance to the agrochemicals to get in contact with a non-target organism. The third area in orange here, looking at using nanotechnology to improve biointeractions. And here a good example would be to make pesticides, for instance, to be uptaken more effectively by the target pest, while perhaps avoiding uptake by the non-target pest. Yeah, I'm going to stop there. So you can go to the next slide, John, please. There are other applications as well where nanotechnology may help achieving new goals, and I'm going to just give you some examples of things scientists are thinking about. The first point is looking at plant nutrition as a complement to plant protection. Has been really wonderful examples, some based in the US, with copper oxide and zinc oxide being shown to boost the immune system and resilience of a number of crops. So this is really the interactions between nutrition and protection, trying to take advantage of what lies in between. The second point is looking at using nanoparticles to increase the resilience of crops to abiotic stress, so drought, salinity, temperature differences, for instance. Here there are some good examples with selenium oxides, for instance. And the last one looks at biofortifying crops, and that's probably more relevant for staple crops, or let's say a type of cereal, for instance. There are some soils that are deficient in some elements that are very important for human nutrition. And so there are a large part of the population that may be deficient in copper and zinc. And the applications of nanoparticles of copper and zinc have been shown to increase the content of these elements into the grain, making the diet more balanced for the consumers. Now, I would like you to go back to the slide where you were. Yes. And the last example is also, again, probably more relevant for staple crop. It looks at those situations where the soil is contaminated with things like cadmium, lead, arsenic, but the land should be used for food production because there's a lot of population there, and the applications of some nanoparticles has been shown to reduce the uptake of those toxic contaminants into the crop. So basically making farming safe, the production of safe food on some soil that are contaminated, and without the use of nanotechnology, the food would be contaminated, which is obviously highly undesired. So you can go to the next slide now, John. So this was the overview of what people and mainly scientists have shown so far. And I'm going to pass the stage to John, who is going to tell you more specifically about the relevance of nanotechnology in wine growing. We cannot hear you, John. You're
Yeah.
muted. Yeah,
Okay,
here you
there we
go.
go. All right. Hello there. Good morning. So my name is John Habito, and I'm a fourth-year student from the School of Environment here at the University of Auckland. So I just want to share what I've been studying for the past 3 years, and I am studying the potential applications of nanomaterials in wine growing, and I have a special focus on the environmental fate of nanomaterials and also their potential ecological effects. So my research is funded by the Borgato Research Institute here in New Zealand. So they're doing a lot of research in wine growing and wine science. So I'm very excited to present to you the findings of my research. Okay. So my research question is, what is the current state of development when it comes to applying nanomaterials as pesticides in vineyards? So so in order to achieve that, we conducted a literature review, and in order to make that, in order to make an objective assessment of their suitability, we used a problem-oriented approach. So we first identified the five major grapevine diseases, considering the difficulty in managing them, the possible economic losses to vineyards, and also the proportion of published research dedicated to developing new methods to address these five diseases. So these diseases in ranking are powdery mildew, botrytis, a grapevine trunk disease, downy mildew. These first four are caused by fungal pathogens, while number 5 is caused by a virus called the grapevine leaf roll associated viruses. So in order to, again, assess their suitability, we decided to disassemble these priority diseases into their problem-associated challenges. So first and foremost would be pesticide resistance. So it's the development of basically pesticide resistance in these grapevine pathogens. So that basically reduces the efficacy of the current pesticides we're using in vineyards over time, and they do not work as well as before. Say, okay, the second problem would be no pesticide efficacy. So this pertains to grapevine trunk diseases, number 3, and number 5, grapevine leaf roll diseases. So current control methods are limited to just cultural controls. So for in terms of grapevine leaf roll disease, the only way to sort of control this disease is by identifying and removing infected grapevines and also controlling its insect vector. And for grapevine trunk disease, another problem is that it's caused by multiple causal organisms. So there's a range of fungal species belonging to different classes that that can cause these diseases that could cause this disease. Okay. Another problem that's common in the use of conventional pesticide is environmental contamination. So basically, a lot of the pesticide inputs that wine growers are applying on grapevines do not actually stay on the grapevine. They're actually washed off or degraded quickly by environmental factors. And again, this sort of goes into the problem of the challenge of limited protective duration because the conventional pesticides that are applied have very limited environmental stability. So they're again prone to mobilization from the side of application, and also breakdown from environmental factors. Okay. So again, because of these factors, they do not confer sufficient plant protection to cover um, the grapevines to prevent these diseases. Okay. So here's a graphical representation to summarize the process of matchmaking the priority diseases and also the nanopesticides. So here I want to highlight again the problem of no pesticide efficacy when it comes to dealing with grapevine trunk diseases and also with grapevine leaf roll disease. So for conventional pesticides, no conventional pesticide can offer any solutions. But when it comes to nano starts to present some solutions for us. Again, going more into detail of the cultural methods used for grapevine trunk disease, it's basically applying pruning wound protectants after pruning, and also over application of conventional pesticides over long durations. So again, in terms of nanopesticides, they're showing novel modes of actions that aren't exhibited by conventional pesticides, and therefore they can provide solutions to managing these diseases. And again, in terms of development, again, there's a whole range of nanopesticides available, and that shows great promise in wine growing. But the development of these nanopesticides are uneven. So so some of these nanopesticides are already in advanced development. That means that they're already conducting multi-year trials, and most notably lignin-based nanocarriers over in Germany. There are also nanopesticides are in the early stages of development, so they're in lab scale or greenhouse scale trials. And there are also a range of nanopesticides that are being applied on other plants, let's say tomatoes and cucumbers, that show great potential when we apply them to wine growing. So these include like nanocopper, nanosilica, nanokitesun, nanosulfur, which is the main nanopesticide of interest for my research. And I think these nanopesticides and their novel properties will be of great interest among wine growers. And of course, if I have time at the end of this presentation, I can go into detail of some of these nanocarriers and how they work. Okay, so I'll go back, pass the mic back to Melanie and she'll talk about the behaviors of nanomaterials in the environment.
Thank you very much, John. So at this point of the presentation, you know what nanotechnology is about and what it may be able to do for wine growing. And in the scientific field, we keep reading really excited researchers promising a revolution. This is not really where we are at, but we believe that there is a really good potential to do better what we've been doing so far. And what I would like to talk about now is the potential and intentional consequences of this nanotechnology, which is all about how they behave in the environment and how they may have negative impact on non-target organisms. So non-target organisms are all the things that is not the pest that you're trying to control, and this includes human, aquatic, and terrestrial organisms. So John, if you can go to the next slide. I want to give an overview of the non-target organisms we are caring about. And for this, I'm taking the regulatory risk assessment framework that every agrochemical needs to go through before it is authorized on the market. Every country has slightly different ways of doing things. Some jurisdictions are more stringent than others. But the idea is always the same, is to make sure that we understand what happened to the substance that is applied, understanding, predicting where it's going to end up, and trying to understand how toxic it may be to this non-target organism. So a typical human and environmental risk assessment would consider 3 stages: the mixing loading, where the main non-target organism is the person actually manipulating the pesticide that is highly concentrated. Then we have the application stage, where the pesticide is applied into the field, and there are very, very high probabilities that some of it, and sometimes a very high proportion of it, escapes the target area. And this is where non-target organisms, both terrestrial and aquatic, may be exposed to the substance that is being applied. And then there is the after application stage. Here we are thinking about the workers entering the field after application to be able to harvest the grapes, for instance, up to the consumers, either the raw product or transformed product, which would be the wine or just the grapes in our case. And so there are lots of studies, laboratory field studies that are typically being conducted to test a new agrochemicals to make sure that it's not going to lead to dramatic consequences on non-target organisms. And so you can go to the next slide, John, please. And so if we go back to the changes that nanotechnology may imply, on the previous slide, you may think that you've seen this slide, but it's actually a different slide. The first slide, I was showing what advantages can be. So for instance, an improved application or an improved biointeraction in terms of the target pest. So we're going to be more efficient at killing the pest, or we're going to reduce the losses to the nearby stream. All these changes may actually have unintended consequences. So if we take the reduced losses, for instance, we may be thinking about an active substance, I don't know, an insecticide, a pheromone, let's say, that it degrades really, really quickly, which makes it unsuitable for application. So we're going to associate it with a nanocarrier, which will make it to be more persistent. If this substance lasts for an extended period of time, it means also that it may accumulate on the leaves, on the grapes, on the soil. And if this substance is toxic to non-target organisms, this is going to increase the risk for unintended consequences. Another example that I'm going to take for an improved biointeraction, that's quite an intuitive one. If the uptake into a certain insect is increased by a nanoformulation, there are really high possibilities that the uptake by beneficial insects, let's take honeybees, for instance, there are high probabilities that the uptake by these beneficial insects may be increased as well. And so we may increase our impact on the environment. And so we really want to avoid these things. And a lot of people are it's kind of a very polar debate sometimes, where people in the room are saying, oh yeah, nanotechnology is doing everything better, and another pool of people say, oh yeah, but they are very risky, they are going to be associated with increased risk. I'm somehow in our team, we are somehow in the middle, trying to develop tools to investigate those processes and provide data that is going to help us develop nanoagrochemicals that are really going to maximize the benefits. So for instance, to address issues that are not currently addressed by current solutions, while making sure that the negative consequences on our ecosystems and on human health are really minimized. So John, if you can go to the next slide. Here, I just wanted to give you some examples of the laboratory work that we typically conduct. These are some series of tests to understand what happens to the agrochemicals after it is applied in the field. You see this leaf here? This is to represent some tests that we show to assess the rain fastness or the resistance of pesticides from rain, from washing off from the target application site into the soil, where they may be unwanted and where they may expose non-target organisms change the microbiome of the soils, for instance, and things like this. We study the degradation of those substances in soil light exposure. We sometimes run microcosms where we are trying to understand how the nano formulation may change the distribution of a substance between the root and the shoot, or between the soil, the liquid phase, the water that is in the soil, the microorganisms, earthworms, and so on. So these are the kind of tests we conduct in the laboratory. And because I've been working on the applications of nanotechnology for a number of years, we applied a lot of those tests to a lot of nano agrochemicals that were provided by collaborators around the world. And John, if you change to the next slide, this is just to give you an overview of the diversity of nano agrochemicals our team has been working with. So on the left-hand side, you can see copper. Copper is relevant for wine growers, so this is why I thought I would mention this example. We conducted fate studies and toxicity studies on different types of copper formulations, including quite experimental nano formulations where copper oxide was loaded on silica nanoparticles or on graphene oxide sheets. And we observe significant differences in how available the copper would be, for instance, and how toxic it would be. On the right-hand side, this is the work that we conduct on what we can call organic active ingredients. It's not organic farming at all. These are classical active substances that are used to control insects, weeds, or fungal disease. And there are lots of scientists in the world trying to make their delivery being more precise using nanotechnology, designing nano carrier that may be made of a range of polymers. Sometimes they are derived from so there are biopolymers such as zein, which is extracted from corn. So trying to really make green nano formulations that are going to increase the efficiency and efficacy of the active substances that are already on the market. One reason for researchers to use active substances that are already on the market is that because the risk assessment process is so stringent, it's very, very difficult to place new active substances on the market. It may take 10, 15 years of work, very big investment to demonstrate to the regulators that a new active substance is not representing unacceptable risks. And so there is a lot of really pragmatic advantages as well to consider the reformulation of existing active substances. The regulatory process to place those new formulations on the market could be much faster. Still, it is not easy, and regulators, follow all the processes and due diligence to make sure there are no unintended consequences, but they were probably looking at 2 or 3 years against 15 years if it was a new active substance. So there's a lot of work on this. So if you want to go to the next slide, John. This is when I'm going to give back the stage to John, who is going to share the type of work that is conducted specifically on nanosulfur. Thank you for your attention so far.
Right. So for the next part, I will be discussing about my PhD research, which is the specific application of nanosulfur in wine growing. And again, I decided to, with a special focus on its environmental fate. Okay. So my main research question is, what are the novel properties does nanosulfur exhibit? And how does it compare when we use it to bulk sulfur? So again, a bit of a background. So bulk sulfur is around 3 to 20 microns in diameter. That's the particle size. So while nanosulfur can be 1,000 times smaller than bulk sulfur. So again, bulk sulfur is being used in here in New Zealand, in Australia, and I know they're using it on the East Coast in the US. Okay, so in the early stages of my research, I wanted to determine whether nanosulfur exhibit these novel properties. So first is improved rain fastness. So I wanted to assess whether nanosulfur exhibits this novel property. So for and I wanted to do it. I wanted to determine whether it exhibits this property when we tested it on grapes and also on grapevine leaves. Okay. So basically, Melanie has mentioned before that rainfast is basically a measurement of how well a pesticide sticks to the plant surface in response to wash-off. So that can come from rainfall or irrigation. So, and for this set of experiments, we were interested in how it performs against bulk sulfur. Okay. So we tested different types of nanosulfur with different particle sizes. So that's 60 nanometers versus 30 nanometers in diameter, and also nanosulfurs with different surface coating. So that's nanosulfur with stearic acid coating and nanosulfur with a polyvinyl pyrrolidone coating. So here are the different sulfur formulations when after preparation. So notice the difference in color of nanosulfur compared to bulk sulfur. So our rain fastness test basically consists of spraying these sulfur formulation respectively on detached leaves or on grapes, and then we dip them in artificial rainwater to simulate wash off, and then we quantify how much sulfur is in the wash off and compared to the sulfur remaining on the leaf surface, and also comparing that to the sulfur absorbed into the leaf tissue. So here's what the different sulfur formulation looks on the surface of the grapevine leaves. So these are SEM photographs. Again, you can see differences in morphology when we look at them at the scale. Okay, so this graph basically represents the distribution of sulfur residues in the wash-off. That's the leftmost bar. On the surface of the leaf, that's the middle bar, the yellow one, and the green bar on the rightmost represents how much of the sulfur was absorbed into the leaf. So here we see that different nanosulfur showed improved rain fastness compared to bulk sulfur. So that's about 19. That's about nine. Oops. That's about 19 to 33% more than bulk sulfur. And with regards to the proportion of sulfur left on the leaves, there's not much differences in between the groups. And just to highlight that the nanosulfur also showed improved leaf absorption. So that's about 25 to 35% than bulk sulfur. Okay. So for the grapes, it's a different story. Like there's not much of an improvement. Let's say about a 10% improvement only in rain fastness, and we saw that in the nanosulfur with the stearic acid coat. However, we did see like an improved leaf absorption in the nanosulfur, stearic acid coated, and nanosulfur PVP coated compared to bulk sulfur. Okay, so for our succeeding study, we want to determine if the improvements in rain fastness translated to enhanced efficacy when we're managing powdery mildew, and we did this through a small field trial. So we conducted a field trial in collaboration with the Connecticut Agricultural Experiment Station and the School of Environment, and again, this was funded by the Brigata Research Institute. So here we wanted to test the different nanosulfurs that we tested in our rain fastness. Okay, so here's the range of measurements that we did for our field trial. So we measured of course we wanted to measure efficacy of the nanosulfur and whether it inhibits the symptoms of powdery mildew. We wanted to see if the nanosulfur treatment causes any phytotoxicity or stress to the grapevine. So we did this through oxidative stress measurements and lipid peroxidation. We also wanted to see if there were any changes in nutrient content in leaves and grapes due to the nanosulfur treatment. And of course, we wanted to measure whether it impacts fruit yield and also the enological properties of the grapes, which I think will be highly relevant to winemakers. So This field trial we conducted for 56 days, starting at bunch closure, at which there were no observable symptoms of powdery mildew in the plots that we performed the experiment. So here I just want to highlight that we applied nanosulfur at 10 times less the application rate of bulk sulfur. So we did 3 spray applications, one at day 0, day 22, and day 41. And all treatments included a baseline treatment of captain. That's basically to control for downy mildew. So we didn't want that to confound our results. Okay, so again, this graph represents the disease severity of powdery mildew among the different sulfur treatments. So here we can say that nanosulfur treatment overall exhibits enhanced or similar efficacy to bulk sulfur. So we measured about a 6% improvement in efficacy and we saw that in nanosulfur stearic acid coated. And again, I want to emphasize that nanosulfur was applied at 10 times lower the application rate of bulk sulfur. So here it is showing that it has enhanced efficacy. So in terms of nutrient content, here it's expected the sulfur content was much higher in the bulk sulfur because we applied at 10 times less the application rate of bulk sulfur. But interestingly, we saw a reduction in sodium content in the nanosulfur treatments compared to the bulk sulfur. Okay, so if you look at the nutrient content in grapes, nanosulfur did affect sodium, potassium, and silica content. So we saw reduced sodium content in the nanosulfur treatments, similar to the leaf treatments. There was some slight increase in potassium content in the grapes, and also some variability in the amount of silica in the grapes. So in terms of enological measurements, so we measured degrees Brix, titratable acid, and pH. So nothing much happening with regards to the sugar content. With regards to titratable acid and pH, we did see nano specific effect of course nanosulfur. So in terms of titratable acid, we saw lower titratable acid content, and also with regards to pH, we saw that the pH was less acidic in the nanosulfur treatments. And in terms of fruit yield, we did not see any differences in fruit yield. But again, this is only in the context that we started the spray application at the bunch closure stage. So that's when the grapes aren't enlarged yet enough to see that tight configuration of the bunch. So at this stage, it's probably too late to see any effect of the nanosulfur treatment. But again, it's still possible that if we apply the nanosulfur treatment at the start of the growing season, then we could probably see improvements in fruit yield. But again, we would probably need to run another field trial for that. Okay. So in summary, nanosulfur shows promising properties when it comes to managing powdery mildew in vineyards. And again, and here surface coating plays an important role when it comes to the performance of the different nanosulfur treatments compared to bulk sulfur. And so this is going to be a very important consideration when we're going to formulate the nanosulfur for when it comes to developing a formulation suited for wine growers. And again, in terms of the properties that it showed, okay, yes, we showed improved rain fastness, we saw enhanced efficacy, changes in nutrient content. Basically, it looks like the nanosulfur didn't cause any stress to the grapevines. No fruit yield in the context of the field trial that we did, and some effects on immunological properties. But again, nanosulfur still has a long way to go before it can be widely accepted and widely used by wine growers. And there's still a lot of knowledge gaps that need to be addressed in order to achieve that. So first would be the ecological impact on non-target organisms. So as a last part of my research, I'm currently um, studying the effect of nanosulfur on the soil environment, looking at its effect on earthworms. There's also knowledge gaps in terms of environmental and human safety. And again, we need to gain a mechanistic understanding of how nanosulfur affects the grapevine. So seeing why it affected the grapevines in our field trial. So we can do this by measuring gene expression or also looking at its effect on the metabolism of grapevines. And again, most important, what will be the effect of the nanosulfur treatment on wine quality? So specifically on the sulfur-containing metabolites in wine, which instill some unique wine flavors in especially with regards to white wine. So this can be like formation of hydrogen sulfide that generates rotten egg flavors, or thiol or mercaptans, which imparts some unique flavor profiles in white wine, like flintiness or gunpowder or struck match. So, yeah. I'll switch back to Melanie again for the.
Yeah, thank you very much, John, for sharing all these exciting results, and thank you for balancing the pro and cons and highlighting that more research is needed. I think this is very often what researchers conclude, oh, we need more research. But I think the results that you've shown, showing potential efficacy at 10 times lower application rate is really promising and exciting. And I hope we'll be able to continue research in this direction. So this slide, I wanted to wrap up and remind you the key messages that we wanted to convey today. The first point, the first part of this presentation was an overview of the applications of nanotechnology in agriculture. And we saw that there is a really great variety of potential nano agrochemicals. And John shared with us how nanotechnology could potentially help addressing the current challenges that wine growers are facing, which includes the control of some disease that are currently not controlled at all with classical chemistry. So there is really potential applications to improve wine growing. There are also discussions, which is out of our scope today, but discussions for wine making as well. The applications of nanotechnology in the food system really goes from growing things up to producing things and preserving things. And so, yeah, if you're interested in these other parts of the system, there are information available. What we do in my lab is not only looking at the positive aspects and the potential benefits of those technology, but we want to make sure that there are no unintended negative consequences. So we think of the potential implications. And this is not to be against applications of nanotechnology. It's more to try and guide developers in designing new nanoagrochemicals that are truly more performant. So that are, for instance, they wouldn't be only treating a disease, but killing all the bees in your plot, but really trying to get the best of all. And to achieve this, we need to work together, not only among scientists, but also with practitioners who are actually in the field using those potential products in the future. And this leads me to the next point where I highlight the need to really compare carefully those nano formulations with existing products. And this is something that sometimes is forgotten in the literature. We know that there is already a lot of diversity in different formulations of copper, for instance, and this is important to use those as benchmarks for nano products to make sure that the benefits are really worth it to place, for economic benefits, to to put a new product on the market, but really thinking about the benefits that the users may get in taking a new product. And the last point is really to reiterate that the work of John on that focused on nanosulfur suggests that nanosulfur may be really promising, but we shouldn't forget about potential negative impacts. And John is currently working on it. So if you want to hear about this in the future, John should be able to share his results in a couple of months. So this is it for us today. We want to thank you very much for your attention, and thank you very much for inviting us to share what we do. This is really important for us to communicate and disseminate our results to people outside of the university. So thank you very much for inviting us today.
Thank you, Melanie. And John, that was really, really interesting stuff and exciting work taking place. You know, a lot of what we've learned today gives me hope about the future and how we can do more with less. And so we're going to open it up for questions right now. I'm looking at the chat box, trying to see if there are any questions raised at this point. Give me a moment. We can also ask questions audibly. Please raise your hand. Use that function. If there are questions within the crowd, if there are no questions, then that is dignifying that you guys did a really, really effective job, and we appreciate it. And give it another minute here. And I would love to stay in touch just personally via your contact info, and that's there for everybody else on the screen. So I don't see questions.
Yes.
We will.
That's a very good point, Eric. Thank you very much for mentioning it. So my email address is on the last slide, and please feel free to contact us if you have questions or suggestions. We really are open. So please do not hesitate to reach out.
All right. Well, you guys enjoy your weekend. Thank you again for making the time, and we learned a lot.
Thank you for the
Good one.
invitation.
Until next time. Bye-bye.
Thanks. Bye-bye.
And for the group, thank you all for joining today. We're going to wrap up a little bit early, but that probably won't disappoint anybody. Hopefully everything was exciting and novel and all the things we strive for every year. I know it was for me. And we also hope that you guys sign up for next week. So if you haven't, please consider that. Also consider aligning with Farm Bureau, joining Farm Bureau if you're not a member, encouraging your employer to join Farm Bureau. And until next week, have a wonderful weekend. Christina, do you have anything to add?
No, Eric, thank you so much, everyone, for joining us today. We so appreciate you being members of the Farm Bureau, and we hope you enjoyed everything. Thank you so much, Eric.
Thanks, guys. All right, signing off.